Okay, great. Thank you. Well, good morning, everybody, and welcome. My name is Steve Thompson. I'm the current chair of the Invasive Species Council Board of Directors, and it's a real pleasure to welcome everybody here today, both here and those that are joining us online for this Invasive Species Research Conference. I'm delighted that it's being held here in Kelowna. This is my hometown. I was born and raised here. Our family settled in Kelowna in 1896, and we're still farming the same farm out in Okanagan Mission that the family settled on. For those of you that are going on the tour, the field tour on, oh, it's just a bit too. Oh, okay. Is that better? Yeah. Those of you that are going on the field tour on out to the John's property, you'll drive by the farm and you'll see the farm and Thompson Marsh, which was a wetland created by my parents, which were dealing with a fair number of invasive species issues in the marsh. So certainly understand what this is all about. So anyway, so thank you and welcome to everybody. This is a conference that's held every 3 years, bringing together academics, experts, practitioners, and decision makers. There's lots of knowledge in this room, and over the next few days, you're going to hear about partnerships and strategies driving lasting economic resilience. And whether you're here in person or joining us virtually, we've got lots for you. So I'd like to acknowledge that we're gathering here today on the unceded territory of the Syilx Okanagan peoples. The Invasive Species Council BC gratefully acknowledges the territories of the Syilx and the Okanagan peoples, and where we live, work to maintain healthy ecosystems for all. We're honored in all of our work across the province to collaborate with Indigenous communities to jointly address the management of invasive species throughout British Columbia. And so to welcome us today, we're going to have a traditional welcome from Pamela and Grouse Barnes, members of the Westbank First Nation and the Syilx. Special traditional welcome from them. They are both adjunct professors at the UBC School of Nursing. And they're joining us this morning just before they have to go teaching shortly after this. They're going to give us the welcome and then leave. They're members, as I said, members of Westbank First Nation. They're honorary fellows of Okanagan College, and they facilitate cultural teaching in partnership with Kelowna Heritage Museums, the Kelowna Art Gallery, the Rotary Centre for the Arts, and regional parks. Pamela's academic background is in Aboriginal government and in the true Syilx tradition. She strives to educate and mentor the next generation in all the work she does. Grouse is one of the fluent Nalexian speakers. I probably didn't say that quite right, but I'm sure he will give us the lesson. Dedicated to preserving and revitalizing the language. He serves as an elder in residence at School District 23 in Okanagan College and works with the Okanagan Nation Alliance on traditional lands management. In February of 2024, Pamela and Grouse were awarded the Distinguished Community Service Award by the Central Okanagan Heritage Society. So please join me in welcoming Pamela and Grouse Barnes. Need this or yeah.
Good morning, everyone. Can you hear me okay? Is the mic close enough? Okay. So my name is Pamela Barnes. My ancestral name is Chuchuwasket, which means the coming of a storm or the coming of change. And it's my honor this morning to welcome you to our traditional territory. I want to thank you for the land acknowledgement and take a moment to explain the difference between a land acknowledgement and being welcomed to our traditional territory, because both are important protocols, but they're different. A land acknowledgement is like being invited to your neighbor's home for dinner. You recognize them as your host and you thank them. And it's the responsibility of the guest. We're often asked to come and can you do a land acknowledgement? And I have to say it's awkward because you don't recognize yourself, you don't thank yourself. That's the responsibility of the host. I think the confusion around land acknowledgements comes because history has not been taught fully. Large pieces of the history of what's referred to as Canada, the United States, British Columbia, and Washington State have been omitted. That is being corrected, but we have a ways to go. And so most people are not aware that formally there have not been the formal changes between ourselves that have been here for thousands and thousands and thousands of years, and those that have come to these lands in the last 200 years. Those formal steps have not been taken. And just because people push through the front door doesn't mean that they're owners or have jurisdiction or any of the rest, no matter how much they say so. The formalities have not taken place. And most aren't aware of that because history has not, the telling of history has not reflected that. So I think that's where the confusion has come in. So that's what a land acknowledgement is. Being welcomed to our traditional territory is also an important protocol. And the first thing I want you to know about it is that it's not a casual thing. I want you to understand that it's important that it is a Syilx person welcoming you to our traditional territory, not just any Indigenous person, because all peoples that have come to these lands in the last 200 years are visitors or guests. And so it's just not appropriate for anyone else to do that welcoming. Just like, if the President of the United States were visiting Canada, we would expect the President to recognize Canada as the host, thank Canada for their hospitality, but it would be an overstep if the President then went on to welcome others to Canada. We would recognize that as not appropriate. Same, in our traditional territory. The other thing I want you to understand is that I want you to understand what our lands mean to us. Our lands are made up of the blood and the bones of our ancestors for thousands and thousands and thousands of years. It is all sacred to us, not just one place over here and another over there, all of it. And when we're out on the land, we're mindful that we are literally walking on the blood and the bones of our ancestors. And we have that kind of reverence. In our teachings, we're reminded that any one of us, we're just here for a moment in time. And the way that we look at our relationship to the land, the water, to all the life that it sustains, we look at it as we borrow it while we're here from future generations. And that's a very different concept than what has come here in the last 200 years, where the focus is on human ownership of the land. And I use this necklace to help us understand the profound difference between these two worldviews. If I own this necklace, I can sell it, trade it, give it away. It's mine. I can do whatever I want with it. I can take really good care of it, or I can just toss it aside. It's mine. I can do whatever I want with it. But the moment that I borrow this, everything about those relationships change. I can't sell it. I can't trade it. I can't give it away. It's not mine to do that with. Even more importantly, there's an expectation that I take the best care that I possibly can, and that when I leave it or return it, that it's at least in the same condition that I found it. It's with this understanding of our sacred responsibility to the land, to the water, to all the life it sustains, and to the future generations, that we first welcomed others about 200 years ago to our traditional territory, even though they were not invited. And it's with this same understanding of our continued responsibility to the land, to the water, to all the life it sustains, and to all the future generations, that I welcome you today to the traditional, unceded, and currently occupied territory of the Syilx people, with the reminder that none of us, including ourselves, we are not doing the best job that we can to care for the land, the water, all of the life that it sustains, for all of our children, all of our grandchildren, all of our great-grandchildren, many of which we will share. We're going to be at some of the same weddings, some of the same baby showers, some of the same graduations. When are we going to figure out this relationship? And when are we going to figure out this relationship with the environment? Lim Lim.
Going to be a hard act to follow. Like Syilx Quest. Hello, good morning. What a bunch of smart-looking people. Isquist. My name is Grouse. My ancestral name is Keenan-Dum-Dan-Quetston. It's about that long. My dad had a sense of humor. How you interpret my name is bumped by sister-in-law. So there's a long story behind that. Anyway, you know, when I hear my bio being read, holy smokes, I want to meet that guy. Anyway, I would say a little prayer. You guys don't have to get up, just relax, enjoy. I live no cool and shouldn't can wish. Dolly has these post can a lot that Syilx Quest. God's quiltum some apanashi Syilx Quest. Dolly has kill yatswit. It's me still that's got kin. Me cap stimmies and kailsted. Why happen a white tikulamitum hula? White skitskish kaipsus, yat stim pelal. Insan at least naksil putikuala, putiku kuikels makmak, kukaus tikum, kukaus witsum, kakulum, pecham, yat stim, at skulstum iltum hula. And that's calm now, sispilki isa herbs can calm truck, putti alai stilchal, putti hali siulk, putti alai itemich now. Why apana why tali hast schal. Thank you, Creator, for allowing me to say a few words and a little prayer. You know, the things that you guys are going to be talking about today and next couple of days is so important. You know, when I pray too, I pray 7 generations ahead of us so that water still be clear, you know, that fish still be here, the animals and the trees. You know, we're not doing a very good job as human beings. If even down the valley here, you look at all the animals that are red listed, you know, we're not doing a good job. So we got to think about the future. And it's not only for my people's future, It's for everybody's future. The things that you're doing now today, tomorrow is so important that we know how to combat these invasive species. I believe even humans are invasive. You know, if the animals had it their way, they would send us to Mars, I think. Then we'd invade somebody else's space, right? You guys have a good day. Thank you.
You gonna do the language lesson?
Yeah,
Oh, shh, shh.
I feel power here. I'm a language teacher. What's Okanagan Lake famous for? Pogo is his or her nickname. The real name, I still got good eyes yet. If you don't try it, I'm going to point you out. Nachachaitk. Okay, listen. Nachachaitk. One more time. Last chance. Nachachaitk. It's better. First part of that word is chacha, which is sacred. Second part of that word is itk, which is water. Sacred being of the water. Ogopogo came to be in 1924 through the Kelowna Board of Trade. And my interpretation of Ogopogo is, come, spend your money. There should be a sighting pretty soon. One more. The simple word for water is Syilx. Try that. Syilx. A little louder. Syilx. I'm going to point somebody else.
Yeah,
First,
might be
first part is Syilx. Humans drink out of the palm of your hand or from a glass. The second part of that word, Syilx, is the sound animals make when they lap the water. So it's humans and animals together, not one more important than the other. Things to ponder. You guys have a good day.
Lim. Thank you. Have a great day.
Well, thank you very much for that very warm welcome. I'm not sure I think I would really enjoy being in one of their classes, although getting pointed out might be an interesting part of the class. But again, thank you to Pamela and Grouse for their very warm welcome here this morning. Before we move to our first presentation, I'd just like to call up Nick, who's our senior research officer with the council, to just deal with a couple of housekeeping and logistical issues for the day in the morning.
Thanks very much, Steve. Yeah, just a couple of notes for people in the room and online. In case you're wondering, bathrooms are just out that door and around the corner. There's also a variety of cafes and sort of restaurants open on campus at the moment. There's a Tim Hortons, there's a Starbucks, and another cafe just out front there. All of our presentations are happening in this room over the next 2.5 mornings, I guess. Lunch will be in that building across the way. That is the administration building. It's in the sunroom is where lunch will be. And then our reception about 5:30 later today will be in the UNC ballroom, which is a little bit further over there. We've got some maps which have all the buildings, and there's stuff on the website as well. Let's see what else. In terms of the online attendees, so you're going to need to move between sessions. So once one ends, it's not going to automatically bump you. So make sure you follow along and click on to those new sessions. You're unfortunately on mute, so you're not going to be able to ask questions live, but you can use the Q&A function, and staff here will reinterpret the questions for the speakers. If you're experiencing any technical difficulties online, please go ahead and message one of us to help out. We've got the research program. There's a couple QRs posted around the place, so you can get that. Oh yeah, sorry. So yes, here's just the pictures of clicking on your next sessions. Would also encourage the folks online to go full screen. So there's a little arrow you can click on there. And I'm sure it will be fine, but if we need to muster in any case of an emergency, we'll just head down the stairs out that door and we'll meet out in the courtyard out front. We do have first aid kits. Lindsay, her hand up, has first aid, so hopefully we won't need to see her in action. And yes, final piece, if you do want Wi-Fi, you can log on to the public one, UBC Visitor, and it should just let you in there. So I will skip us ahead one more time, and I will let Steve introduce Carla. Steve.
Thanks, Nick. Just a note before I introduce our first speaker, some of the presentations over the next 3 days are eligible for continuing educational credits with the BC Landscape and Nursery Association. We will let you know which of those presentations are eligible, including this presentation. So if you're collecting those credits, please fill in your details on the log sheet at the registration desk to get your credits. So it's my pleasure to introduce our first presentation today. We're very pleased to welcome today's keynote speaker, Dr. Carla D'Antonio. Dr. D'Antonio is a distinguished professor at the University of California, Santa Barbara in environmental studies and ecology, evolution, and marine biology programs. She teaches fire ecology and restoration ecology at UCSB and conducts research on post-fire landscapes, as well as the restoration of native species to degraded landscapes in California and Hawaii. Prior to teaching at UCSB, Dr. Antonio worked in the USDA's Exotic and Invasive Weeds Research Program and was on faculty at UC Berkeley for 13 years. She has worked in Hawaii, California, and the Great Basin. During the past 38 years, Dr. D'Antonio's research has focused on the ecology of invasive plants, their influence on fire regimes and post-fire recovery, as well as community structure, ecosystem processes, and the success of ecological restoration. Very, very appropriate topic for us and for our province. So I'm very pleased to welcome Dr. Carla Antonio up, and we look forward to your presentation. Thank you very much for being here.
Thank you so much. And thank you to the organizers for inviting me here. It's really an honor to be invited to speak here and a pleasure to also learn from all of you what you're working on and what your concerns are here. So I look forward to the next couple days. So before I begin, I want to acknowledge that the work that I am going to talk about is the result of many, many interactions and collaborations with the people who've been in my lab all these years and a whole range of land managers throughout the US and funding sources. So I was thinking back when I was asked to speak here to the very first keynote address I gave, and it was at the 1992 first annual California Exotic Pest Plant Council meeting, which became the Invasive Plant Council. And I recently moved offices and I found a manila envelope full of my handwritten notes from that meeting. And I looked at what I emphasized. And the questions I emphasized were things like which invaders could really transform ecosystems? How do they do this? And what can we learn about them that would help us exploit their vulnerabilities and better manage our ecosystems? So it was really a call for research and the need for research on these topics. Because back then, there really wasn't very much published research on invasive plant species. And the red arrow here indicates 1992. And you can see there weren't very many publications. So it was a cry for more research. And since then, there's been a tremendous amount of research, most of which is single taxon papers shown in the light gray there on invasive plant species. And a recent publication by Monsivilla's group in Spain shows a similar thing across the European literature, increase in number of publications on invasive plants. But pay attention to that light gray line there. that's the increase in the number of invasive plant species. So there's also been a steady increase across this time in the number of invasive plants. And it isn't just that we become aware of them and so we list them, it's that there are actually more and more of these spreading across the landscape. So in the 33 years since I gave that talk, we're seeing this proliferation of research, but we're also seeing a continued expansion of invasive plants. And in fact, in the environments where I've worked, such as Hawaii, California, in particular, I've seen these plants increase their footprint across the landscape. So research and publications, single taxon publications in particular, isn't really helping us get to the answer to controlling and managing these species. So more research on invasive plants is simply not enough. This is part of a figure from a paper published in Frontiers in 2017, focused on translational ecology, which is defined as the interaction between practitioners and researchers in defining and co-producing questions and knowledge. And that to really move the field forward, we need to focus on that intersection and not simply on our favorite research questions or practitioners just focusing on how to kill plants. So I'd like this talk to be a contribution to the call for action at this nexus of research and practice, but with the not just on invasive species per se, but invasive species as part, or managing them as part of ecosystem restoration. And in particular, focusing on multi-species communities rather than single species studies and multi-value systems. And I think you'll see a little bit more as I go on what I mean by multi-value systems. So in a very simplistic sense, what we are trying to do when we manage the abundance of an invasive plant species is move in the direction of greater functioning or achieving the values that we have decided we want from ecosystems. And we don't have to necessarily get rid of them to increase the value or function of the system. So some of the themes that are going to come up through my talk, which is aimed at what we have done throughout or the kind of research I've been involved in. I just realized I didn't turn off my phone. Sorry. So one of the themes is that removal of a single species can release a secondary invader. And this is well covered in a recent paper that I was involved in, published also in Frontiers. This was a pandemic project that a group of us, we were supposed to have a meeting in April of 2020, and we didn't have that meeting. So we met on the internet and we devised this paper addressing this problem of secondary invasions, where you remove the first invader and a subordinate invader is released. Or if you remove both invaders simultaneously, the legacy of one of them promotes the other one. So we cover this in great detail. And it's been a theme of my work that when you start manipulating species on the landscape, the species that respond aren't necessarily the ones you want. So secondary invasion really has to be considered. For example, in Hawaii, one of the first invasive species removal projects I got involved in was trying to manage fire-promoting grasses. And when we removed them from the ecosystem, we found that they were replaced by a worse grass, a more flammable grass, and a tree, a nitrogen-fixing tree that really filled in the system. The dark bars in this graph show the increase in the number of those invasive species in the ecosystem compared to the control plots. A second theme of my work has been reinvasion by the original invaders is likely when you try to control these ecosystems. So for example, an ecosystem we have in coastal California and that we've been involved in trying to restore in the campus area at UCSB are called vernal pools. These are shallow wetlands that form on a clay hardpan that exist only during the spring, late winter and spring. And we restore them by removing invasive species, which are one of the main problems in them. But one of my students, Joanna Tong, recently published part of her dissertation work showing that when you stop managing them, which happens usually 3 to 5 years after the beginning of the project, so when you stop removal, they come back. And here you see 3 different color lines, the upper one being the outer edges of the pool, and that's where the invasive species return the most intensely. But the transition zone in the middle in green also shows an increase in invasive species. Only in the pool center do you get just a small increase. And that becomes important because it helps us think about how to do restoration, how to maybe change the structure of the pools physically to have better habitat. But the theme here is that you have to expect that the reinvasion by the original invaders is likely, unless you have some great biocontrol. And the third theme of my work has been that native species need help. They are often seed limited, especially relative to invaders. The timing of arrival is important. You need to get them into the system fast because you have a short window of opportunity to get them in. And seedlings face many challenges and may need a lot of help. And I won't go into too much detail on that stuff today because there's a huge literature on restoration that addresses these things. But basically, to enhance the desired functions, you need to control secondary invaders. You need to control reinvasion. You can't just walk away. And you need to assist more desirable species in colonizing to aid the successional process. So these things seem pretty straightforward, but when you're working on the ground to try to restore a system in the face of massive invasion by non-native species, you usually find yourself really frustrated. And some colleagues of mine in Hawaii put together this article that I think is a nice read if you're interested in this problem and have faced decades of trying to remove non-native species. And so I'm going to steal from them the terminology of sort of when can you beat them? Or do you not beat them? Do you give up and walk away? And so I'll address, using examples from my work, when can you actually beat them? What do you do when you can't beat them? And how do you keep going forward in the restoration and management of invaded systems? So I want to start with some examples from California, focusing on a species that I did my dissertation on in the 1980s. So I've been working on this plant for more than 40 years. And it's Carpobrotus edulis, or we call it highway ice plant. It was brought into California in the 1800s, late 1800s, early 1900s, for control of sand along railroad tracks and then later along roadways. It has spread widely in coastal ecosystems, and it invades multiple community types from the dunes going inward to the bluff and the coastal scrub, and then grassland wetland type communities. And those different community types offer the ability to understand how community structure and other species interact with a non-native species. And what we see if we think about resistance to invasion is that in the dune, invasion is actually fairly slow because it's an abiotically harsh environment, whereas in the bluff scrub and the grassland wetland type habitats, there's no resistance. These plants just, once they're in there, they just take over. So, but we do have a resistant sort of sage scrub community. So what do we do when we have an environment like this? So this is an aerial view of a reserve where we've been working recently, and everything in the yellow and orangish color is an ice plant individuals. This is a massively clonal plant taking over the landscape. And many people now are mandating in their agencies that people remove this plant. So can we actually do it, and can we actually win? And I want to give you an example of where I think we have won and why we've won. So this is the UCSB campus. For those of you who aren't familiar with it, it's probably most of you. And what we have here is a system that's relatively limited in where dispersal can come from. So I don't know if you guys can see the pointer here. No. Okay, well, the campus area, where we're in one of the natural areas where we've been managing, is surrounded by the campus. And this plant is an obligate animal dispersed plant. There are no deer coming out to this campus point. So there's limited reinvasion potential. We know from aerial photos that prior to the Navy being here in the 1940s and 50s, there was a vibrant coastal scrub community there. There's today, so we know we have a community to aim for that's resistant. There's very little disturbance, and there's high community engagement, meaning lots of people who want to get involved in restoration, like 26,000 students. So we have devised a methodology there using black plastic to kill the ice plant, because we don't want to use chemicals around all these students and around the campus lagoon. And with 3 months of coverage by black plastic, we effectively kill the plant. We restore with plants grown by the students. We within 2 years, we have a vibrant coastal scrub community. So we remove, we restore, and we steward. We steward by repeated visits by campus classes, by all sorts of community groups, by the local Indigenous Chumash people there. So we have a really active stewardship program, and that helps maintain the site as a really vibrant site. But the vegetation itself also helps maintain the site. So we can beat them because of the isolated location, because we have a resistant community to restore to, because we can control large-scale disturbance, and because we have high community engagement. And in fact, this project is one of several really successful projects we've done on campus or in the campus area. And I would say that it makes me much more optimistic about the role of restoration in semi-urban settings, the role of controlling invasive plants there, because it's this huge educational opportunity for the local community and people to be involved and to connect with nature and to help you with that reinvasion problem that you're often going to face. So sure, you don't support big carnivores or wildlife there, but you really have a lot of other positives about these kinds of settings. Okay, so let's stick with Carpobrotus just briefly here and talk about a much more challenging situation. And this is the reserve managed by the Nature Conservancy, an hour and a half from campus. And my PhD student, Nick Saglambeni, is focusing on how to manage ice plant. Here, the Nature Conservancy has a mandate by the California Coastal Commission to remove 1,000 acres of Carpobrotus. Okay, that is a super optimistic thing. I can honestly say, having worked on this plant, this system is it's part of a 40,000 acre parcel that's adjoined on multiple sides by the military and by private ranchers and a state park. So completely open borders. There's Carpobrotus everywhere, and I see this as this open system. Where can you possibly be successful? And we're actually having a meeting at the end of this month to address what to do about this. The area is large, it's super windy, traditional control is difficult. The black plastic approach was just ripped to shreds in a matter of weeks. Even chemical control is hard because of the wind at this site, but you can do it. And we found that repeated application of chemicals is necessary. So going back to this habitat gradient that I introduced, where can we be successful? And actually the colors here should be indicative. Green, we're successful. Red, we are not successful. And let's look again at where we can be successful and why. So we have, Nick, I should say, but with my help and his committee, over 300 acres of experiments They're trying to manage this plant and build resistant communities across this environmental gradient. So in the dune, we can be successful because nature's helping us. It's an abiotically harsh environment. The regrowth of the plant is really slow. Seed influx is low. A lot of the seed dispersers don't come out to the super windy dune edge. And there's limited secondary invaders because of the harshness of the habitat. So we can kill this plant and keep these habitats pretty free with a little bit of stewardship after the initial kill. So removal alone is pretty much enough here to restore the system. And here's an example. So this is 1 year after removal. There's Nick walking through the sea of native species, mostly Abronia umbellata, that came out of the seed bank after we killed the initial plant. So really successful with almost nothing else other than the removal itself. So that's like a great thing. We wish it could happen everywhere, but it doesn't. So let's go to the coastal scrub community. Here's another place where I think we can be successful, but it takes a lot more effort. And here we have to beat them with restoration. We can't just remove. We have to stay at it. So for example, what happens, you get a lot of secondary invasion. The orange line here, the black line is Carpobrotus going down. Orange line is invasive grasses coming in. And the other thing that happens is the reinvasion by Carpobrotus from root fragments and from the seed bank. So resistance is critical to create, and we have to do it quickly in this window of time after the initial kill. And to get good cover, we found that seeding is better than planting, and that's great news, but seeding is really low diversity. So seeding, planting yields much higher diversity, but it takes a lot of people and effort. And we do get UCSB students out there, but there isn't a local community to support this effort. So we're really going to probably go mostly with seeding to try to get cover as quickly as possible. Cover is important not just for competition, but because we have found that animals eat the seedlings. And so we need to get the cover to get the animals in there to help eat the seedlings. And we also have found that isolated shrubs, and this was actually part of my original dissertation work, are overgrown easily by regenerating plants. So to really generate resistance, we need that clumping in the background you see at high density. And so again, seeding is what's going to give us that kind of high density. So we have a plan for getting to resistance there, and we're moving successfully, I think, towards that. But it takes a lot of effort. But what do we do in these other habitats? Do we just give up? Do we walk away? There really is no resistant community to build. So for example, in the bluff scrub type habitat, the indigenous plants there are very low growing. They are not competitive. They don't shade out the ice plant. They don't provide a lot of habitat for the herbivores that will eat the ice plant seedlings. So we end up, if we try to restore this habitat, it just gets reinvaded and overgrown. So we either have to invest massively in constant stewardship or maybe try to figure out some kind of combination of species. But it's really an expensive investment, and we are facing the issue of giving up, but just trying to protect the few places that are not invaded yet. So focus on protection of what's left and not on restoration of what is already degraded. And I think this is a really, really common realistic problem that are faced in this world. And we're struggling with that one. Now with the grassland wetland communities, here we use black plastic. We actually use a whole variety of things to kill the plant. And the problem with this ecosystem is secondary invasion. So we pull out the ice plant, we see this massive invasion by a grass, Festuca perenni, and this thing is super, super thick and persistent. So different ways of killing the plant on the right show secondary invasion with the orange bars. So what are we going to do? So we're proposing that we actually live with the ice plant in this habitat because Carpobrotus edulis is much better than Festuca perenni at supporting a diverse pollinator community. It's actually a great pollinator plant. And it provides a lot of habitat for herps and burrowing animals, much more so than Festuca perenni. But there's one other alternative that we're going to recommend to the Nature Conservancy and see what they say. And that is to start over and change the goal. Okay, these are the same kind of shallow habitats that form these shallow wetlands that I told you about in a previous slide with the vernal pool habitat. So we're proposing that in some of these areas, they actually kill the ice plant and then go in and scrape out and create pools, create habitat that's unfavorable abiotically for Carpobrotus and gives them a whole different set of values. So there's a lot of interesting species in vernal pools that we could make habitat for by simply giving up on the idea of restoring the original grasslands there, because Carpobrotus will reinvade. But if you make a really wet pool, it won't reinvade because it doesn't like to be wet. Okay, so there's some other long-term challenges. So I've shown you we have habitats we think we can restore with a lot of effort. We have habitats we can restore without too much effort. And we have habitats we just don't know what to do with. But we also have the fact that there's no public access and there's limited Native American access to this site. The Chumash peoples managed this land for 10,000 years. Now the Nature Conservancy has a total hands-off policy. I view that as a real negative in terms of ecosystem restoration because people interacting with land in a regular practice is going to be, I think, critical to restoration. The Nature Conservancy also manages this land as a ranch as part of their sort of agreement when they bought the land from the ranchers. But livestock on this property degrade resistance to invasion. They get into those shrubland habitats and beat them up. They also eat the Carpobrotus fruit and spread it. And the area is surrounded by other ranches. So we need some kind of regional strategy. So this just points out the sort of regional level problems you have trying to control within one area when you have a plant dispersing in from all over. So I want to shift gears a little bit now that I've shown you sort of what do you do when you are facing these big challenges and talk about other approaches we've taken to dealing with widespread problematic species, but still trying to find some success. And I want to shift to talking about invasive grasses in Hawaii. So invasive perennial grasses in Hawaii are real game changers. They are things that change fire regimes. They stall and hijack succession. They reduce native plant diversity and structure, reduce habitat for indigenous species. They are really big landscape changers. So there's no realistic control methods over the large areas that they cover. Reinvasion is absolutely a certainty. They disperse quite easily. Secondary invasion is problematic in some areas. I already showed you some slides from my early work in Hawaii there. Native resistance is quite low, and fire is inevitable in some of the invaded areas. So what have I done besides frustrate myself for all these years, right? I've sort of shifted to work with managers on a very local scale to find out what can we do that would give back to them some of the values that they want from these ecosystems or that we all would like to see. So we focused on, at a local scale, how much grass reduction or what level of control is needed to reduce fire risk and the impacts of fire on those systems. What level of control is needed to promote succession and to allow natives to coexist in a matrix of non-native species. So we're focusing more on coexistence rather than total getting rid of non-native species. Oops, I don't know what happened there, but that's not supposed to look like that. So I'm going to focus first on the dry forest example where we have fire being a major game changer. So this is a system I told you about earlier. And once it burns, it converts a diverse woodland environment into basically an African savanna environment dominated by one grass species. So what can we do? So this is work I did over a 15-year period with Hawaii Volcanoes National Park managers. One thing you can do is try to keep fire out. So identify what are called special ecological areas, diverse woodland or shrubland environments, create green breaks around them, close down the backcountry, and hand weed in isolated areas that are sort of buffers to keep the weeds out of the more critical areas. So that special ecological area project is being driven by the park. Then in the degraded sites, oh dear, wow. Sorry, I'm going way too slow, I guess. We've used prescribed fire to create a window of native re-entry back into these communities. We've basically accepted that we can't go back to the original community, but we can create these designer communities using native species that tolerate fire and live with the grasses. We've conducted 9 prescribed fires, seeded with 11 different native species, some of them shown here. And we've created these communities that don't look like the original community. Here's the original community, but they have native species in them. They create a heterogeneous canopy that slows down the spread of fire, and they support some of the native birds. So we can't go back, but we can go forward. And we have to think creatively about putting things in there that will tolerate the new disturbance regime. Now, I just end with this one example at the high elevation forest we've been working in recently on Mauna Kea. And we've asked what level of control is needed to promote succession. These forests were cleared for livestock grazing. Hawaii has huge private ranches. And after the Fish and Wildlife Service bought this land and set it aside and got rid of all the cattle and the sheep and the goats, it did not undergo succession to native forests. So they went in and planted a native nitrogen-fixing tree, but that tree promoted a non-native grass species that became quite abundant in the understory. So we've been looking at how to set succession in motion by removing these grasses. So we've planted native species in low and high density, and we've also herbicided. And I want to show you these herbicide results because it's a secondary invasion problem. If we use herbicide to get rid of the grasses, we get massive invasion by rubus, which is a really worse invasive species. So whereas if we just plant at high density and we don't herbicide, we get grass reduction on its own. So total removal of grasses promoted the secondary invader. So we've decided to let native woody species do the work. We need to plant at high density to mimic the kind of recruitment we see in the intact forest, but it requires a lot of plants and a lot of people. But we have that, okay? We have a huge commitment by the local community. There are so many community groups, mostly indigenous community groups, Native Hawaiian homelands people who want to be involved in restoration here. We have regular groups volunteering to come up there and working in the greenhouse there. And this is how we are doing a successful restoration project there. We accept that our native woody species will be living amidst a sea of non-native grasses, but we feel that we are moving the system in a direction that people care about. I'm going to skip this graph in the interest of time. So the solutions may not be a fully native community or even focusing just on removing invasive species, but rather on what values can you put back in the system and how do you get natives into them. Whether it's this fire responsive community that we've created in Hawaii Volcanoes or this clumped native community we're creating at the high elevation forest site. So I want to end by zeroing back in again on the importance of working with managers to understand what they want, but to help them also understand what's possible in a site and to focus on multi-species ecosystems where we can improve the values in some way. And I'm going to end with a quote by a Canadian. Okay, and that's Eric Higgs from the University of Victoria. For restoration to be successful, people must be involved. So thank you very much. Okay, do we have time for questions?
I think we do. Yeah, I think we do. So we've got time for a few minutes for questions. So anybody like to be the one to start us off with some questions? Come at the back there.
Okay, great. Yeah.
Sorry,
It seems to be that what you're describing is gardening at a landscape scale, which I don't, that's not an objection. That's I'm being enthusiastic about it. I love gardening and think it's great. My question is, when you're doing sort of the reseeding, for example, post-fire, what are the values involved in choosing only native plants? Given that you've said that there are non-native plants that behave themselves quite well and might add ecological value.
That's a great question. And in fact, that article that I referenced at the beginning, the beat 'em, join 'em, or start over, in that study by my colleagues at the Institute of Pacific Island Forestry, they actually went out and did a huge trait analysis of native species, invasive species, and invasive or non-native but not invasive ones, and then generated communities that included mostly natives and non-invasive non-natives, right? And it was really a trait-driven choice of species. Now, we didn't do that at the Hawaii Volcanoes Park project because the park really objected to anything that was non-native, even if we could prove it was not invasive. And to be honest, most of their problems in the park, or most of the plants in the park, are either native or they are invasive, meaning harmful, super abundant non-native species. So we didn't really have that choice in that situation, but you bring up a really good point. And I think for a great example of it, you should look at that article that I cited, because they generate these really rich, interesting communities. And they've also surveyed the Native Hawaiians, and they really value those communities because they contain plants that they value for either food or wood production that are not native, but they are not invasive. Some of them are species they brought that we call canoe plants that were brought when the Polynesians settled in Hawaii. So I agree, we don't have to stick with just natives if you can achieve the values you're looking for with non-natives. Yeah, behind you.
Yeah, I get the trait-based approach. I think I can see the merit in it, but I'm really curious what you think about co-evolutionary relationships and complexity, because that's where I start to wonder how important being of a place is over time, as opposed to just selecting a suite of traits we need for ecosystem functioning.
So your question is really about what do I think about the potential loss of co-evolutionary relationships? Well, I'd say that we've been losing them pretty steadily over the last couple hundred years to begin with. But where we can preserve them, we should preserve them. But in a lot of the situations I'm working in, it's a choice of going down a pathway of total dominance by really undesirable species, or trying to shift the system towards at least some more desired functions. Some of these things that we're putting in the system do, like in Hawaii, some of the native plants we're introducing, they have insects that live on them that are specialized to them, that have evolved with them, like psyllids that occur only on particular species of Hawaiian plants. And those relationships come along with them. So I think that, we've kind of focused on
what we think of as sort of foundational species of the woody plants that we're putting into those systems, and then hoping that the co-evolved relationships that they have with microbes or with insects will come along with them. So we're not losing everything. And I feel like the other situation of not doing anything, we lose everything. And going back to the original communities is just impossible. It's really an impossible feat in so many of these systems. So I appreciate, your point, but I feel like our choices, at least in the places I've worked, are so sort of dire that, we are willing to maybe sacrifice some of the co-evolved things, but I'd say they've already been sacrificed. So, yeah.
I'm really curious about,
Yeah, I'm really curious about sort of the case study you had talked about where it seemed like potentially the best option was to switch goals altogether to like a vernal pool creation. And in general, if you have insights into at like what point do you decide to pivot, especially I'm thinking about this a lot in the context of climate change too and restoration. Thank you.
Yeah, I don't have an answer there at all. At what point you decide to pivot. And actually, I'm kind of looking forward to our meeting with the Nature Conservancy I guess it's actually early June that we're having that meeting, because I don't think they're expecting to hear this. I think they're expecting, like, they've supported Nick's work there and they expect us to give them a recipe for how to kill ice plant over 1,000 acres. And you just can't believe how much regrowth there is and how much problem there is with that. And so to have us say, look, there's this whole region that we can delineate where you just need to kill it and then scrape it out and create a totally different habitat. I don't know what they're going to say to that. I think, so I think it's hard to decide. And for some people, that decision is going to be driven by economics. And they're going to have to look at, like, can we steward, like, not there, but at UCSB, we can steward so intensely, right? We can get people out there constantly because we have 90 to 100 students a year volunteering as interns in our restoration program. So we can steward, so we don't have to pivot hugely. But in places like in these vast landscapes, pivoting might be, the only way to go, especially in the face of climate change. So, yeah, climate change is definitely pushing us.
Thank you very much. I just had a quick question. Are you seeing in your research ecosystems that are at higher risk of these massive challenges with invasion? I'm thinking maybe more arid climates and just in the picture of climate change also.
Yeah, I think that's a really interesting question. I feel like in arid systems, like, for example, the dune on that great coastal gradient is our most arid part of the landscape. And I think it's the least at risk, right? Because it's harsh enough that there just aren't very many invaders. And we also saw that in the article I briefly flashed up there on secondary invasions in Hawaii. In the really dry forest, when we pull out fountain grass, which is an invader there, we get no secondary invaders. But in the seasonally dry woodland, we pull them out, we get a bunch of invaders. So I think that harsh environments, you have a more limited suite of invaders. And when you get rid of one, you don't necessarily get the infilling by others. And that the richer environments are much more like the wet forest, where my friends are doing the hybrid ecosystem restoration with non-invasive, non-natives. That's got the most invaders and they're fierce. They grow fast. So the richer environments are almost the more troubling environments. That's not quite true in the ice plant situation because that's where we can build a really good shrubland, and that will provide its own resistance as long as we don't have cows beating it up, right? So, but I think in general, I think that really rich ecosystems are pretty at risk in some of these settings, or the sort of intermediate ecosystems, that the more harsh ecosystems, at least the ones where I've worked, are actually a little more manageable because you have fewer invaders. But you've got to keep disturbance like fire from tipping the system over an edge. I want to just say one last thing about another example of success in the dry forests in Hawaii. There's a dry forest near Kona on the Big Island, so very touristy area that was off limits. It was a natural area with no human access. You know, it was going to be where natural processes take place, right? Kind of an old-fashioned idea. And it got very invaded and degraded. And then the state Natural Areas Commission decided to get rid of the designation of wilderness and wildness and allow human access. And we have all these school groups from Kona going out there and pulling invasive grasses. And the system has come back just remarkably. So it's another system where we don't have secondary invaders. We get rid of the first one, we have people stewarding, and we have success. So, you know, if I leave you with anything, it's the role that people can play, especially in ecosystems where there's not a huge number of invaders to deal with. So, okay, I think I've well exceeded my time.
Yeah, that's great. Well, good. Well, thank you very much. Let's have a big round of applause for Dr. Antonio, getting us off on a great start with a very thought-provoking presentation. I know Carla will be around for the forum here, so I'm sure if you've got other questions or want to discuss any of those ideas with her, she's certainly going to be available. So thank you very much. I'm going to turn it back over to Nick to move it on to the next part of the session.
Just like to thank some of the sponsors that have made our event here possible, and their generous sponsorship is really important. They obviously care about this work and lead in different industries and different contexts, and encourage you to visit the booths and have a chat with everyone here over the next couple of days. So we're gonna kick right into hot topic presentations. We've got 3 speakers joining us today. First of all, I would like to introduce Dr. Philip Weyl. He works for CABI, the Commonwealth Agricultural Bureau International, a nonprofit organization focused on agricultural and environmental issues and the creation, curation, and dissemination of scientific knowledge. He's a research scientist with CABI and leads the weeds biocontrol team, managing projects in Canada, US, Australia, and South Africa. And Dr. Weyl is also an affiliate professor at the University of Idaho. So I think we've got Philip logging in here.
Yeah, I think so. Can everyone hear me?
Loud and clear, yeah.
Perfect. Okay, I'm going to try and advance slides. I hope the IT tech team have done this. There we go. Okay, so I'm going to start back there. Okay, great. Thanks so much. Sorry, we've had a bit of back and forth about how to get the presentation up and running, but it seems like the guys have done an excellent job. Thank you so much for giving me the opportunity to speak to you today. I can't see anybody, so if there's an issue, feel free to just jump in and let me know if there's an issue with the slides, or if you need me to do anything different. So I'll be talking to you today about biocontrol in general, but also particularly for British Columbia, and about the partnership that CABI has with British Columbia, but also Canada as a whole, where we've been working on weed biocontrol for over 75 years. So a little bit of history with the CABI Switzerland Center. So we were initiated in 1948 as a substation of the Commonwealth Bureau of Biocontrol, and this is largely where our Canada partnership began. So in the early days, the center was largely involved in studying forest pests, particularly those native in Europe and invading in Canada and North America. But since then, the emphasis has largely shifted towards agricultural pests and weeds. So this is our center. So we're based in the northwestern part of Switzerland. I'm not sure if you can see my pointer, but the building is up on the left-hand corner. And then we've got various greenhouses, garden beds, some really nice field cages, so we can do some pretty cool experiments. And then we can also do open field tests, which is these sort of crop circles in the bottom right. So we do have a lot of fun in the field season. Okay, and our collaboration is largely thanks to the Weed Biocontrol powerhouse of Canada, which is quite predominantly Western, with Dave Ensing and Chandra Moffat, Val Miller in the picture, as well as Rob and Rose, and then Susan Turner, and I believe from BC, missing from this photo is Janine Brooke, who's also heavily involved in Weed Biocontrol. And it's largely thanks to them that we have such a strong commitment to Weed Biocontrol in Canada. And supporting them from the Swiss side is the team that I lead, which is 5 permanent scientists based at the CABI Switzerland Center, Ivo Tosevsky based in Serbia working on the toad flaxes, and then our technical staff, as well as Harriet Hintz, who is kind of our honorary member past that we rely on for her vast experience. And then every year we also have 7 to 10 research assistants, which are temporary, typically summer students who come over, of which between 3 and 4 or 5, even some years, are Canadian, which is fantastic and keeps that collaboration and connection alive. It is worth noting that many of those past summer students, including Chandra, Dave, Janine, were summer students at CABI. Our collaborative work between CABI and Canada is quite evident from the biological control programs in Canada books, of which we've just published the 6th volume. These date back to 1910 and really highlight the research that CABI and Canada have done, as well as other parties. In the latest book, we co-authored 21 of the chapters between Canadian and CABI scientists. And just a shout out to Megan and Veronica for taking that over from Peter Mason and Dave. Okay, so just to give a little bit of background as to what classical biological control of weeds actually is. So it's the deliberate use of specific natural enemies to control an exotic weed. And that's quite important because we wouldn't necessarily be looking at introducing a biocontrol agent for a native weed. And I'm sure if any of you have ever been in a weed biocontrol session at a conference or in a talk, you will have seen this graph below. And essentially what this is it's a kind of graphical view of what invasion looks like. And very similar to what Carla showed us a little bit earlier, is that we have undesirable vegetation exceeding a threshold level, or damage threshold level. And when they do that, we need to look at management options, of which biocontrol is one in that toolbox. The agent is developed. We only use specific natural enemies, so only those that are targeting the plants of interest and does not affect any native or economically important species in the introduced range are suitable for use. Those are released into the environment. We hope that their population expands rapidly, and that brings down the population density of the invasive weed below this damage threshold, where we might see things like native vegetation recovering, and that may also be helped along with restoration efforts. Okay, so just to put this graphically, what we often have, and Carla, I think, showed this very nicely with Carpobrotus, is that Carpobrotus in the Indigenous range in South Africa is is found almost everywhere, but it's an important part of the ecosystem. So it actually contributes to the biodiversity. You don't necessarily find these vast expanses of monocultures. But once it arrives in a introduced range, plants are typically larger, or they form these dense monocultures, which are problematic for us. And if we consider the top-down pressures, what we often find in both the native and in the introduced range, you may have a set of generalist species. These are typically, and when I talk about species, I'm largely focusing on insects and potentially pathogens, but these are generalists that will kind of nibble here and there, but they don't really have any population level effects on the plant. But different to the native range, from the introduced range, we also have a whole suite of specialist natural enemies, and these are co-evolved herbivores with our plant species that have a kind of evolutionary history and are typically monophagous on those plant species. And those can, in many ways, exert enough top-down pressure to manage those populations. And what we try and do in biocontrol is to reintroduce those in the introduced range to bring the invasive species essentially under control, or to restore that ecological balance. Our goal is to permanently reduce the weed below that economic or ecological threshold that I explained to you earlier. We cannot, and in fact, it's not in our interest to ever eradicate the plant species, but rather to essentially restore that balance. And there's several examples where this has been incredibly successful in Canada. And if we look at some of the historical invasive rangeland plants, for example, St. John's wort, prior to the 1950s, massive problem, covering essentially the prairies were under severe threat. St. John's wort was covering basically the entire landscape. With the introduction of Chrysalina species, which did a fantastic job, you could physically see these populations crashing. So if we look at 1949, this picture here, the yellow in the front of the image is all St. John's wort, and roughly 10 to 15 years later, you can see that invasion is rendered relatively insignificant. And we do have the data to back this up. So what we can see here, and it's essentially exactly that same scenario that I showed you earlier on the graph. We have a high density of the weeds, the agent is released, the agent population booms, and so the plant population crashes. And that's happened across several sites. Another very cool example is houndstang, which is an awful plant. It's got the seeds form these burrs, which will cover. If you take your dog walking through an invasion of houndstang, you may end up with a kind of just lump of seeds and not much of your hound left, which is not great. It outcompetes native plants, forage plants. It's poisonous. Chemical control is limited, especially with the levels of invasion. This biocontrol agent, Mogulones crucifer, was tested and deemed specific enough for release in Canada, and it was released in 1997. And prior to release, so this image here in the middle is the invasion prior to release. Houndstang is a biennial plant, so essentially each rosette lives for 2 years and then flowers and dies. And what we can see is that with the introduction of Mogulones crucifer, 2 years after introduction, the plant, you can already physically see a reduction in rosette density. And 4 years after introduction, those rosettes from that site are virtually gone. Again, rendering houndstang invasion insignificant. And this is not, it's an incredible disperser. This is not only at local sites, this is across the Canadian landscape, which is absolutely fantastic. So what are CABI's role in Canadian efforts in targeting invasive weeds, but not only weeds, but also insects? We assess feasibility of biocontrol, we discover potential agents, we select those those agents and assess them, so essentially host specificity testing. And then we also develop petitions for field release. So our ultimate goal is to get these agents, the risk analysis done to ensure that they're safe, and then to get these agents out in the field. And weed biocontrol typically follows a 7-step, essentially a 7-step process. So I do just want to shout out to Christine's poster later today. In the poster session, I unfortunately will not be there, but she talks about the 7 steps to introducing new agents and the biocontrol pipeline. So if you want to get more details on that, please go and check out her poster. Okay, so in 2025, we have 21 active projects at CABI. 20 of them are for North America. One is co-financed by Australia and one is for South Africa. And most of our projects are financed by a consortium approach. And where this becomes important is that they're typically started by one donors and others follow. And I've really have to have a shout out to the Ministry of Forests in British Columbia here, in that in so many cases, they are one of the first to initiate funding of biocontrol programs, and the others are to follow. And that's something that is incredibly important in terms of keeping these projects going. What I mean by a consortium approach is that we have essentially multiple donors per project, and these ensure continuity and longevity. Some of these projects can be up to 25 years of testing, which is a heck of a long time.
Sorry to interrupt.
And it also means that there's excellent value for money. A lot of these projects, we really struggle to get funding for biocontrol, and we do work on a shoestring budget, which CABI is very good at. But yeah, the kind of multiple donors does help those projects keep going. So I don't expect, this is a long list of our weeds projects, but what I did want to point out here is just the number of projects that British Columbia is sponsoring in terms of weed biocontrol development. We've got 13 active projects within the Swiss Centre and two based in our UK Centre. And just like I was saying earlier, so our sort of new kid on the block, Echium vulgare, which is blue weed, this guy here in the top right-hand corner. That was essentially initiated by funding from British Columbia, and now we've been able to secure funding from the US as well. So that's growing that project, and we'll hopefully be able to make progress much quicker.
Sorry to interrupt. This is your 1-minute warning.
Okay, thank you. If you do want more information, please feel free to reach out. That time went a lot quicker than I was expecting. Have you taken back control? Sorry, I can't go through my slides anymore.
Can you try now?
Yes, I can. Okay, so again, reach out to me if you want more information, but we do have some petitions on the horizon, so keep your eyes open for some agents which are going to be petitioned very soon. This is a list of our agents currently being released. The latest one is Sotranchus constrictus against garlic mustards and Bogus nodulosis. But we do have a whole bunch that are currently being released. And I think that is... Okay, I can't proceed again. Oh, no, there we go. And just to very quickly finish up, so Canada has a very long track record in plant biocontrol. It's safe, effective, agents are self-dispersing, and really economical. And this is largely through our continued collaboration between AFC, and the slide is completely messed up, I'm very sorry. Between AFC with new tools being developed continuously, reevaluating past programs. Through our collaboration, we've targeted 37 weed species, 89 biocontrol agents released, of which 59 are established in Canada, which is incredible numbers. Thank you very much.
Thanks very much, Philip. Any questions for Philip in the room or online? Looks like you may be getting off easy here, Philip. Oh, no, we've got a couple.
That's great. I like questions. It means people are listening.
Thank you very much. Just wondering how widely dispersed when you do a dispersal, say it's in BC, how widely dispersed do the pests go?
So they're biocontrol agents, which are safe, so I don't want to call them pests. It really depends on the species. So, for example, Magullonis crucifer is an incredible disperser, and that'll go kilometers every year. It's being found in really isolated locations, quite distant from any release sites. While other species will disperse maybe a few meters every year. So it really does depend on the agent's ability for dispersal. It is something that we try and look at, but it's really difficult to predict. But yeah, so it is very species specific, and I would say from hundreds of kilometers per year down to meters. So not a very good comprehensive answer for you, but yeah, I hope that covers it.
I'm wondering if you could say something about how you select the agents to invest your time and effort in terms of their impact. Like, you know, we've struggled in the US with agents that are released after a lot of trouble getting the permits and stuff that really don't do much. Like, they establish and they spread, but they make some galls on the plant, but they don't really bring down the plant. So how do you make your decisions about going after agents that are really going to be potent ones?
Thanks. I assume that's Carla, speaking from the US. But yeah, that's a very good question. And then you throw something like climate change into the mix, and it gets incredibly difficult to predict. So it's a very good question, and we do spend a lot of time trying to select agents that are going to be damaging. What we like to do, what we invest fairly early on in a lot of our projects, is trying to understand the weeds biology. So trying to figure out where those Achilles heels are. You know, so some weeds might. You know, for example, the Hans tongue, targeting seed set will work, but over a much longer period. So we really focused initial efforts on rosette feeding or root feeding agents. Where it gets really difficult is, for example, on vines or Carpobrotus would be a good example there. You know, a stem mining insect may break that plant up, but that may actually end up just it may not end up having the kind of impact that you might like. So it is very difficult to predict. We do impact experiments under controlled conditions in the native range, which is why CABI is so nicely situated, because we can do those outdoor experiments, not only working in quarantine, but really understanding the weeds biology and then where your agents fit in terms of damage. So trying to figure out what would limit the weeds population, and then figuring out which agents would attack those parts, if that makes sense.
Alrighty. Well, yeah, thank you so much, Philip, for taking the time and joining us from across the ocean. We're going to hear now from one of your collaborators. Next, we've got Dr. Chandra Moffat, who's a research scientist in entomology, specializing in the biological control of invasive insect weeds with Agriculture Canada in Summerland. Her team focuses on conducting research to develop and optimize biological control solutions for the management of invasive insects and weeds.
in agriculture, rangeland, and riparian ecosystem. Please welcome Chandra.
Okay, good morning. Can you guys hear me okay? It sounds really quiet up here. Yeah. Jason's telling me to grow taller. Okay, thank you so much. And it's great to follow on the heels of Phil's talk. So I'm going to continue talking about biological control of invasive plants. And this is a project that my team initiated in collaboration with the Ministry of Forests about 5 years ago. And I'd just like to acknowledge at the front the immense contributions of the technical team, Tyler Nelson, Christine Koch, and Valerie Marshall, as well as many students, and the Ministry of Forest team, including Janine Brooke and her predecessors, Susan and Sandy, who have done so many of the collections of the insects that we have used in this project. So just following on the heels of Phil's talk and the 7 stages that he mentioned. So the impact assessment stage is the first stage, which is really a co-led effort between the local stakeholder here in BC. It's often Ministry of Forests that are bringing these invasive plants to our attention, their stakeholders bringing them to their attention. And together we work on describing the problem. And then the role of CABI, as Phil was mentioning, is really in the overseas exploration, conducting a lot of the biology and host range testing, and collaborating with us on the petition for release. So with Agriculture Canada, we get involved at the stage very early, but then re-involved in terms of doing some quarantine assessment work and co-developing that petition for release. So our 3 agencies, as Phil was mentioning, really work together, but I wanted to sort of highlight which steps of the process each agency is involved with. But once the agents leave the native range and we have that approval for release, the propagation, the initial field releases, initial establishment and impact monitoring and long-term monitoring, those are a partnership between our science teams with Agriculture Canada and the BC Ministry of Forests. And as Phil mentioned, for more information, you can check out Christine's poster later today on the details of that. But I mentioned that to just provide some context into what's happening here on the landscape here in British Columbia. So this is a picture of me in the field looking at a very persistent invasive species, spotted knapweed, with a lot of sort of questioning, like, why do we have so much knapweed here on the landscape when we've had biocontrol agents for several decades? And I bring this to your attention to illustrate that there's a lot of work that still needs to happen on the ground following the introduction of biocontrols. Because we're looking for agents that are not just efficacious to really reduce the population of the invasive weed, but we're constricted by ensuring that the target weed, that we only bring in biocontrol agents that are highly specific. And so especially more so in the US than Canada, there's an extreme emphasis on safety and extremely low risk tolerance, whereas in Canada, we do use more of a risk-benefit framework, still ensuring that biocontrol agents are specific, but tolerating a small amount of ecological risk. So looking at an invasive species like spotted knapweed, if we want to develop a biocontrol program and manage the weed in the long term, we're really looking at a massive landscape-wide problem. And thinking about where we introduce biocontrol agents and how those biocontrol agents move in the landscape are questions that we continue to address, often decades post-release. So here, this is just showing a bit of a blow-up of British Columbia and the very dense and vast distribution of spotted knapweed on the landscape. So often, there can be many biocontrol agents released for a particular invasive weed. So for spotted knapweed in Canada and the US, about 12 agents have been released, and most of those have established. So as Phil was mentioning, we look to sort of try and find the Achilles heel of the plant. And so for a species like spotted knapweed, it does reproduce via seed. So there's been a number of seed head feeding agents released. But it is a long-lived perennial species, so only targeting its reproductive output directly, it's not the only strategy. So there's also been a number of root feeding agents introduced to really weaken the plant and hopefully kill the plant in the field at times and reduce its vegetative or sexual reproductive output sort of from the ground up. But when we look at these different biocontrol agents, as Phil mentioned, we don't necessarily know how effective they're going to be once they're released. And especially in some of these older systems where basically all of the possible biocontrol agents were thrown at an invasive plant, it takes a lot of post-release research to figure out which are the most successful agents. But in order even to do that, we have to be able to tell them apart. So if you look in this photo, this capitula moth, we have the larvae of that moth. We have 3 different species of tephritid flies, which at the larval stage are totally indistinguishable from each other. And even in the adult stage, it's like venation and coloration on the wings. And the 2 weevil species, I show you the 2 most different photos of them that there are, but I'll show you later how hard they are to tell apart. The same thing goes for these root feeders. So to be able to understand the potential impact that the different biocontrols are having on the landscape, we have to tell these insects apart. And often we're trying to do that when these insects are in their immature stages, which presents a number of problems. So there's a real high need for us to be able to discern amongst the immature stages of the insect, which are completely indistinguishable via morphology, as well as highly morphologically similar adults, like the different fly species I mentioned and their wing venation, and the 2 weevil species that in the field can look basically identical. But another thing we have the opportunity to do is to determine really if we have the right genotypes of biocontrol agents in the right places. And so that starts with understanding really if we have population level genetic variation in the insect populations that have been released and where that is on the landscape, and if we might be able to manipulate that to achieve better biological control. We can use that then to potentially determine which populations of biocontrol agents are best placed in different parts of the landscape. So the goals of this project that we co-developed were to first develop molecular tools to discern amongst very similar, morphologically similar biocontrol species, both at the larval stage and the adult stage. And so to do that, our goal is to develop a DNA barcode library for all the weed biocontrol agents released or that have since come in to BC, which would really allow for us to determine the identity at any life stage without waiting for a lengthy taxonomic identification process. And then our second goal to really ensure that biocontrol agent genotypes are in the right place on the landscape. And to do that, developing a population genomic sequencing pipeline for as many biocontrol systems as we can to determine the level of population genomic variation on the landscape, where agents are best suited, and see potentially if there are any possible hybridization events, which are often talked about as possibly going on in the field. So I'll focus here on these capitula feeding weevils on knapweeds, these 2 species of Laurinus. So these are both seedhead feeders, and even though, like I said, those 2 pictures show them to be very different, they're extremely difficult to distinguish. So I have other photos shown here from a paper by Lukian and Anderson showing Laurinus obtusus, male on the left, female on the right on the top, and Laurinus minutus, males and females. And you can start to get a sense of how morphologically similar these 2 species are. Color and body stoutness are often used to tell these 2 apart, but they're really actually horrible characters for doing so. And so misidentifications happen really frequently. There's so many intermediate individuals in the field, it's almost impossible to know which is which. And to tell them apart, as I was alluding earlier, often requires expert taxonomic identification, which requires only having adult specimens, packing them up, sending them to Ottawa, having them in a taxonomic queue, which can take months, if not years, to get identifications back. That doesn't really allow us to be able to manage the invasive species on the landscape in a timeline that we want to. So maybe before I just go into this further, I want to highlight why we need to tell them apart. So these 2 species of weevils, even though they're so similar, they do have distinct biology, and they are adapted to different parts of the niche. So Laurinus obtusus from the native range is found primarily on spotted knapweed in moist habitats, environments that would be like the Kootenays or the coast here, whereas Laurinus minutus, it's found primarily more in drier habitats, and it's the biocontrol agent while it does feed on spotted knapweed, it's the biocontrol that's had the greatest success in controlling diffuse knapweed, which is a plant in very arid environments. So there really is a high need to be able to tell these two species apart to make sure that they're in the right places on the landscape, controlling the right weeds, and that we can understand the contributions of each to the system to know if we should be moving either of them around. Okay, so we would assume, based on collections from Europe and the original host specificity testing, that these would be two good biological species. So this is just an example cladogram that would show that, yeah, there's a clear split in these two species. But what we found in this messy plot here is an actual cladogram of field identified Lyrinus individuals collected by Ministry of Forest staff, sent to Ottawa for taxonomic ID, and then barcoded. And so in the top part of the plot, all individuals there are Lyrinus obtusus, so showing it's a really good biological species. But in the second part of the plot, there's a total mixture of individuals that were identified in the field and even by the taxonomists as Lyrinus, as both Lyrinus obtusus and minutus, that all, when we look at the DNA barcodes, cluster together. So obtusus is looking like a good biological species, and then minutus, we wouldn't really know what was going on because there's so much discordance between field IDs, expert taxonomic IDs, and the DNA barcodes. So really wondering here, like, were these individuals actually misidentified, or are the barcodes wrong, or is there more going on in the genomics of the species that we don't understand? So to really try and get at this question and better determine what's going on with Lyrinus on the landscape, Ministry of Forests, as well as members of our own team, collected Lyrinus individuals from around the province. We store those in ethanol, prepare the specimens. This is locations where these Lyrinus individuals were collected from knapweed species across BC. And then we use the DNA barcoding approach. So we sequence, we extract the genomic DNA. This is done by students and technicians in the lab, prepare the genomic data and send it for sequencing. And we get the DNA barcodes back. So for these individuals, we ensured that they were expertly ID'd to make sure we know, looking at morphology, which species is which, and then seeing, do the barcodes actually match with the species IDs that we gave. And so when we do this, we actually did find very good separation of Lyrinus minutus and Lyrinus obtusus when we used a combination of lab expert ID and the DNA barcodes. So it indicated that in the earlier data set, the discordance was primarily related to the field ID, so identifying these weevils in the field instead of having the time taken in the lab. But we can, through DNA barcoding, easily distinguish these two species. Then the second approach, which allows us to answer more fine-scale questions, is the population genomics approach. So we use a ddRAD sequencing pipeline. So this is using restriction enzymes to basically scan across the whole genome. So whereas with barcoding, which I didn't mention, it's sequencing like one section of the DNA of the mitochondrial genome, 500 to 650 base pairs, just that like one section of the mitochondrial genome, whereas the ddRAD approach looks across the nuclear DNA in the entire genome and takes little reads at consistent places so that we can compare and have much more data on from the entire genome. So conducting this process, if we had good biological species across the nuclear genome, we would end up with these sort of two ellipses of individuals in multidimensional genomic space. And so when we look at Lyrinus minutus and Lyrinus obtusus, we found we had many more Lyrinus minutus individuals collected, and they cluster very closely together. But there is quite a lot of genomic variation in the populations of Lyrinus minutus, whereas Lyrinus obtusus, fewer individuals, and they're quite similar in genomic space. And we just found one hybrid individual. So these two species do hybridize on the landscape. We have less than 100 samples here, but we might have like a 1 or 5% percentage of hybrids. But we can use the genomic data to definitely easily separate the two species and identify potential genotypes that may be better suited in different parts of the landscape. So just looking quickly at the distribution of the two species, here in central British Columbia, we do find some segregation of Minutus here in green and Obtusus in blue, but often they're coexisting at the same sites, which can indicate that at probably the majority of sites, either biocontrol agent can play a role, but there might be more ecological extremes where only one agent could survive and have an impact. So this is just an overview of both the barcoding and the genomic pipeline. And so just focusing in on this one example study system, the Laurinus beetles appear to be good biological species. The barcodes match the nuclear genetics, which really helps us get at the fact that the barcodes are sufficient for distinguishing between these two different species. They're both fairly widespread in BC. But we also know that knapweeds are doing very well at high elevation. So the genomic data may allow us to potentially identify populations of the weevils that may be better adapted to controlling knapweed at higher elevations. And that's something that Dave Ensing and a student will be working on coming up. So where do we store this data? All of the barcode data gets stored in the Barcode of Life database. This is an example of what our project looks like with the different sequences available and what it would look like for a single species. And to date, we've barcoded just over 45 different insect species, generated 225 barcodes. So this would allow us to be able to address the similar kinds of questions in many different weed biocontrol systems in the province. And with the more detailed population genomic approach, we've been looking at 4 species groups and sequenced about 500 individuals to ask those more fine-scale questions. For 2025, we have a few more target biocontrol agents that we're interested in barcoding and getting those into our database that we haven't yet collected. And I'm a little bit over time, but thanks to the biocontrol team and Ministry of Forests for their partnership on this project.
To what extent do you think that the wide genetic variation in that one Laurinus species is due to evolution in situ since it was brought here, as opposed to bringing it from a wide variety or bringing in a wide variety of genotypes?
Yeah, that's such a good question. And what we often have trouble with is that there's a few individual vouchers that are saved, like when we do a release, like there was only a couple individuals, like 1 or 3 individuals saved. So we don't have that historic lens, but we can start to get at that question, which we haven't done for Laurinus, but to look like how many populations were the original weevils collected from. That would give us an indication, as well as if we know those locations, and other studies have done this, like what is the population genetic variation in the native range and how would that compare to what we have here? But I think like if there's enough standing genetic variation like in the population that's introduced for it to establish, there's enough variation for selection to act on. And when we introduce biocontrol agents into new ecosystems, they're facing a whole new suite of selective pressures. So I think it's really interesting when we see that much variation and trying to address like exactly that question. I don't have the answer, but those are questions that we're very interested in, because it's definitely been demonstrated in other systems that there can be a lot of rapid selection on biocontrol agents when they're first released. That's partly why a lot of biocontrol systems, a lot of initial introductions fail, because selection is too strong for the population to persist. But when they do succeed, there's a potential for a lot of evolution. I will say though, as kind of a caveat with that, it's very unlikely for selective pressure to be able to shift an insect to change its host range though. So there's very few, if any, instances of released biocontrol agents that have ever shown a selective pressure has caused them to change their host range. And for the biocontrol systems where we do have recorded non-target attack on native plant species, that was always predicted in the original host range testing. But the sort of culture at the time of releasing those agents tolerated that level of risk. And I'm telling everyone that, but I'm sure from California that you're well familiar with that, with native thistles. Thank you for your question.
Any other questions for Kendra? If not, we will go international again and to another one of Chandra's collaborators. All right. You gotta. Okay. There we go. Do you know if he's there? Okay. All right. So yeah, next I'd like to welcome Dr. Fabio Malogni from the University of Vienna in Austria, where he's a postdoctoral researcher. His research lies at the intersection of biological invasions, island biogeography, and human geography. In collaboration with Dr. Jason Pither from UBCO and Chandra, the work in his team has aimed to systematically compile and map evidence on the individual and cumulative impacts of invasive plant species. So yeah, please give a warm welcome to Dr. Malogni. And are you there, Fabio?
Yes, I'm here. Can you hear me?
Yeah, can hear you loud and clear.
Fantastic. Thank you very much, and for a nice introduction, and hello to everybody. This project, we aim to collate the existing evidence on the cumulative impacts of invasive species in British Columbia. It's conducted by me, Chandra, and Jason. I'd like to take the chance to acknowledge that this project was conducted in the ancestral land of the Syilx, and I'm thankful to have had the opportunity to live there and work there for a couple of years in this beautiful place. Apparently, there you go. Okay. So background. We decided to focus this project on riparian ecosystems specifically because we know they contribute disproportionately to ecosystem services, biodiversity, and they're also disproportionately impacted by invasive plant species. However, we know that the impacts of invasive species are often investigated in isolation, despite they can interact what we call cumulative impacts. Cumulative impacts have been explored in many contexts. It's not a new idea, but surprisingly, in invasion ecology, research is comparatively scarce. So we decided to focus on two goals. Number one, to develop a reproducible protocol for systematically compile the evidence available on the impacts of invasive species. And secondly, to test this protocol and systematically collate and map the individual and cumulative impacts of 9 plant species invasive to foreshore and riparian ecosystems in British Columbia. A note on a systematic map. Systematic maps are slightly different from systematic reviews. Systematic reviews generally, which probably you're all familiar with, generally have very narrow and specific questions, and they qualitatively assess the literature, at least a qualitative assessment, while systematic maps have only one specific goal, which is to map the available evidence, just to map it and see what's available. So it's a broad question. We wanted to know whatever it's available about the impacts of these species. We use the current gold standards for reporting systematic evidence synthesis in environmental research, or ROSES. We developed first a search string. We decided to develop a string that was purposely broad, so just the name of a species, in this case, Elagnus angustifolia, the Russian olive, and impact. And the idea is to find all the evidence available regardless, without any restriction, or limiting the number of restrictions at the minimum. Then we run these search strings in a set of different databases for literature, like Web of Science, the CABI, and also several from local to international databases, and all reviews we could find. We developed 4 screening criteria. Papers were included if they were about our focus species, about the impacts of the species, within the reference habitat, so riparian and foreshore ecosystems, and within North America, which we defined as only Canada and the US. Next, we screen the papers. So first, title and abstract, if that is sufficient, also the full manuscript, and we extracted information from them. We developed this protocol iteratively using two species, the Russian olive and the reed canary grass. So we started with these two species to develop our protocol and our workflow and our search criteria. And then after extracting the paper and screening them, we extracted also the data. Which data did we want to extract? First, a description of the impact, their magnitude, and their directionality. In particular, we used a well-known tool, the ICAT, which define impacts based on, quantifies impacts based on the impact that they have on biodiversity. So an impact can be, for instance, minor if the changes in biodiversity affect only the fitness of individual, or moderate if it changes population densities, major or massive if there are extinctions. But ICAT doesn't account for direction. So this is useful for magnitude, but we also wanted direction. So we use a slight variation of the ICAT, which is the ICAT plus. The ICAT plus includes also the direction. So if the change is a reduction in fitness of individuals, then the impact is negative. So it's just the absolute value, the absolute change. So we define as negative or positive an absolute value in the fitness of individual, the population density, or the number of species. So for instance, it could be positive if there's an increase in population density or the introduction of species, of new species, native species. Results, we publish the protocol, which is available. It's a flexible protocol. It's open access, can be applied in other contexts and on other species, and you can check it out online. But more than that, we also completed the screening and the mapping for, partially the mapping for 9 priority species invasive to riparian ecosystem in British Columbia, 2,654 studies screened. 92 studies that we included, so about 4% of the total of the literature we screened. And we identified from these 92 studies 312 impacts. But these 92 studies are not evenly distributed across the board. As you can see, some species like the reed canary grass and Russian olive comprise of almost 2/3 of the studies that we identified, while other species there are few or no studies, like common tansy. We found no studies investigating the impacts of these species at all. And similarly, and somewhat unsurprisingly, the evidence is increasing over time, but there are gaps, geographical gaps. So the south, the north, and the middle of North America are virtually, we have no work coming from these provinces and states. In general, a surprising result is that directionality of the impact is largely negative, but we have a good chunk of impacts, 30%, which are positive, and almost a quarter that are relatively small. And then regarding the magnitude, we have again a good, almost over half of the impacts are negligible or minor, while only 25%, 27% are major or massive, whether positive or negative. Then I present this slide. I will present now 4 different graphical framework, or better, the framework is the same but for different species. And there will be circles associated with ecosystem components. In this case, for instance, you can see the fish, and there's a circle for the fish, and there's a number in it, which is the number of impacts we actually that were investigated and we extracted from the literature. And the colors within the circle indicate the directionality. Again, yellow is positive, blue is negative, and white is no direction, is a negligible impact and the magnitude. So the darker is the color, the more negative, or the more positive. So from minor to massive. And here, the Russian olive, for instance, you can see how the literature have investigated impacts on almost all ecosystem components. Vast majority is on birds, but also bats is actually represent all mammals in this context, but also plants and terrestrial invertebrates. For the reed canary grass, again, we can see there are impacts all over the graphical framework. All components, almost all components have been investigated. But the focus here is largely on soil property, soil fungi, and soil communities, and plants. Well, if we move in the Tree of Heaven, for instance, we start to see large gaps in the available evidence. And again, a strong focus on plants and soil properties. This is not surprising for Tree of Heaven, is of course related to the allelopathic effects of this plant, which have been well studied over the years. But for other components, we have almost no information. And the same goes for the Himalayan blackberry, where we have a few studies on birds, but other than that, the information starts to get a bit sparse and scarce. And for the other species, remember that the Common Tansy was not produced 0 articles. For the other 4 species, the yellow flag iris, giant hogweed, wild parsnip, and Himalayan balsam, there were only a few studies, and they investigated almost exclusively 1 or 2 ecosystem components. Most of the impacts were negative, and mostly referred to plant richness and diversity, or effects on arthropods and macroinvertebrates. In conclusion, we have 2 species that were investigated extensively. The Russian olive and the reed canary grass. There are large gaps for basically all the other species. The evidence increases over time, but with large gaps geographically. Impacts on other plants other plants are the most investigated, but there are other ecosystem components that are neglected. Here I mentioned fish because of course its cultural importance in BC, but also lizards were almost not investigated. I think there's only 1 single study across the board, across all species. And of course, we need to expand to other invasive species, such as the knotweeds and Phragmites, which are next in line in this project. I take the chance to thank you for listening, and thank you, say hello to everybody who knows me in the room. And also, I would like to remind you that Jason is gonna talk more about this project in a couple of days, on the 15th in the morning. He will work on, he will present a project on, while here I presented the evidence we have, he will present if this evidence is reproducible, and focusing on, again, Russian olive and the reed canary grass. Thank you for your attention.
Hey, thank you very much, Fabio. Any questions? All right, a couple for you. We're just running a mic here. 1 sec.
Yep.
Yeah, thank you, Fabio. Amazing talk. I'm curious, in evaluating the impacts, in addition to focusing on gaps in specific taxa, have you considered also looking at like what types of species interactions have been most studied or less studied in terms of the impacts of these invasive plants on species interactions? Thank you.
So yeah, thank you for this question. It's actually surprising how we have little to no information about the interaction between the species, whether the interaction is between the non-natives or with the other species. So we don't know exactly these impacts. Actually, this is a pretty big gap that we don't know how the impacts of these species end up interacting, if they are addictive, so they sum up one with the other one, or if some species actually, the impacts of one species reduces that of another one. We are planning to work on that and to try to present the results in a way that help us at least visualize what might be happening, but we really need some proper work to understand what's going on, because we really don't know. Generally, what we have are papers that just explore maybe 3 or 4 species, and they just tell you which are the impacts of these species, but not the interaction between them. We just assume that it's all negative and it's all and there's no interaction between them.
Okay, thanks, Fabio. Another question coming your way here.
Hi there, Fabio. Thank you so much. It was very interesting. And just for context, I work in local government, and I was wondering if this type of research would be of use to potentially prioritizing the eradication of certain invasives over others due to those impacts, like you were highlighting with the tree of heaven, just in recognition of limited resources, or sort of in light of our keynote talk speaking to needing to do ecosystem restoration instead of the focus on eradication. So if there's certain ecosystems that we could focus on for restoration, it'd be of value to sort of prioritize which invasives have the greatest interactions through your kind of research.
Yeah, I tend to share the view that not always eradication or leads to the results we want, and sometimes restoration might lead to more invasive coming in. But yeah, the idea of this project is to actually inform the ministry where we should focus our attention, to understand what are the impacts, but especially where we should focus next in terms of research, to understand if species like, I don't know, the common tansy actually have impacts, and which are the impacts first. And then when we know what are the impacts and what are the interactions between these impacts possibly, we can start focusing our effort in one or the other direction, because in some cases it's going to be very difficult. If you think about the tree of heaven, it's very hard to manage these species. So we want to make sure that we are actually not just doing management or eradication based on a hunch or how we feel about a species. In some cases, there are species that have been investigated extensively, but always by a couple of research groups. And that shows a bias. So we want to avoid this sort of situation. We want to make sure that when we go for a species, we do it with as much information as possible behind our back.
All righty. Well, thank you so much, Fabio, and thank you for taking the time to join us from Austria. All right, so we are now going to go into a little coffee networking break. Sorry, we're a bit behind here, but it's all going to work out in the wash throughout the day. Yeah, unfortunately, we've had one speaker not able to join us later today, but so it'll all work out. But anyhow, thank you very much to our sponsor, CD3 Systems, who's sponsoring this next coffee break. And yeah, we'll be back here in 15 minutes to get cracking again. So yeah, thanks very much, everyone. And oh, sorry, please give a warm round of applause to Fabio.
Thank you, everybody.
All right. Good. Good. There's a request from someone online I was chatting with. So there are people asking questions if they would interact, like say the people online can't see who they are, say like who they are, who they work for. Yep. Should I say? Yeah. So if anyone like types in a message, then whoever's reading it Yeah, the online people were hoping that people in the room could say that I'm so and so because they just realized that
Talk in the next session.
You transfer it over to the USB drive? Because the
You want
problem
it there? You couldn't do it online, or what's better?
Miranda.
Okay, yeah,
The best part is if you do a USB drive, we're gonna merge it here.
Okay.
But the problem is we can't disconnect the laptop.
Yep, yeah, I've got a dongle in.
Oh, perfect. It does say waiting for Fabiano to control your screen, but it doesn't give me an option to terminate the control or to stop share, but that might actually be the screen share.
Yeah. Weird.
Yeah.
Yeah, because I guess even as a co-host. Oh, weird.
You should have the control now. Sorry, I forgot to click the button.
Okay, perfect.
It's on.
All right. Good morning, everyone, and welcome back. Thank you so much to CD3 for supporting our networking break. Super appreciate having your help putting on this conference and having you here. If you haven't checked out their booth, please go say hey to Mark at some point today. All right, my name is Lindsay Barnett. I'm the manager of planning with the Invasive Species Council of BC. Super happy to be here today and have all of you folks here with us both in person and online. I'm here to introduce the next session, which is called Prevention. And we have 4 presenters for this session, so we'll follow a similar format. Each presenter will have 15 minutes, and then we'll follow up with some questions. And our first presenter today is Dr. Miranda Hart. Dr. Hart is a professor of biology here at UBC Okanagan, and her research focus is soil biology and conservation of microbial, communities. Please welcome Dr. Hart presenting biofertilizers, invasive species underground.
Have a thing?
Yes.
They should work. Just fine.
Okay. Okay, thank you. I'm really happy to be talking to this audience because I feel that microbes are left out of invasive species research. Maybe that's just me. I'm not looking around enough, but yeah, I don't know. I think things are changing. I think things are changing really quickly. Do you really need, do I need to do this? Yeah? No. Okay, okay, fine. I don't know if you guys have noticed this, but fungi are having a moment in the popular culture, which is a long time coming for me. Thank goodness. I don't know why this is happening. It's kind of perplexing to me why people who don't, I mean, you don't have to convince me. I love fungi, but I'm always surprised when it just permeates popular culture. This is an article that was in the New York Times, March 1st. And like twice a year, New York Times are running long length articles about fungi. It's crazy. This particular article was about how networks, fungal networks, coordinate and grow. I mean, interesting. But I'm bringing it up here because I want to come back to it, and it has to do with threat of invasion. This is from the paper. There's so many authors. It's a group from the Netherlands who do this. Actually, one of my former PhD students is in this group. We found that mycorrhizal fungi build networks as self-regulating traveling waves, the density of which is self-regulated by fusion. Okay, whatever, they're growing in waves and they fuse. And that's really, really important. And I'm going to come back to it. Okay, here is a network, a hyphal network. And this is also from the same group. This is from the New York Times article, but it's also on their website. What is a mycelial network or a hyphal network? I have a video, but I don't know how to use things that aren't Macs. So Just, it's like a video. But I don't see the button. Maybe it's in the next, there we go. Okay, so this is real time. They have a robot at the Vrije University in Amsterdam, and they're looking at these artificial systems, but hyphal networks, right? And so these are basically just mycelium with all sorts of stuff, you know, nuclei, compounds, whatever fungi are synthesizing or uptaking, and it's transferring around the network. And you can see things are moving forwards, they're moving backwards. We don't really understand yet why or how they move and what coordinates that movement, but we do know that a single genotype, a single fungus is connected to many, many different plants in a landscape. And a single plant can be connected to many, many different fungi. So in this way, it's an elaborate web in every ecosystem that's joining above and below ground communities. Okay, and people are getting excited about these networks. Uh-oh, okay, wrong way. Okay, how do these networks pertain to fungal invasion? And where's the problem? Well, the problem is here, global biofertilizer industry. And this is the bulk of my research program, looking at these products, testing these products, and worrying about the threat of invasion from these products. Now, biofertilizers are really essential. They're just microbes. They're microbes that are mostly commercialized. They're produced commercially, and they're sold to farmers, restorationists, to improve plant performance. You name it, they're promising that biofertilizers do this. It's growing really quickly. It's worth about $3 billion, the industry in the US right now. In the next 10 years, it's going to be worth 3 times that. And it's growing so quickly because these are marketed as green alternatives to like synthetic pesticides and herbicides and fertilizer. So it's growing really quickly. It sounds good. From the outside, it sounds good. However, if you look inside what these products are, yes, they're microbes. Yes, they're not harmful chemicals, but they're single genotypes and they're produced in really artificial conditions. And we all know here what happens when you introduce a single genotype into an ecosystem. I mean, sometimes not a problem. Sometimes it doesn't establish, and sometimes it's a really big problem. So this is my fear. And my fear is because they're global, these genotypes are being globally distributed. They're not just local. They're going, for example, the one that I look at most closely was isolated in the 70s from Rivière-du-Loup in Quebec. And has become the superstar of the arbuscular mycorrhizal fungal world. It's distributed everywhere in the world now. One single genotype. So we've been studying this genotype really, really carefully. But we know, ecologists know, soil plant ecologists know, that soil isn't just one genotype. It's many genotypes of fungi, thousands upon thousands of genotypes in any system. And these communities of fungi change dramatically among plant communities. So what's growing with this Western red cedar in a forest in central BC is very different than what's growing near Syilx. And it's very, very different from what's growing in a vineyard, which is one of the systems I look most commonly at, right? There's fungi in all of them. There's networks in all of them. The fungi are very, very different. I'm focusing on the fungi because that's what I study, but you can just apply this to all the microbes down there. Okay, soil, microbes, and plant communities. This is a soil core. We look a lot in my group at what fungi are there and how human disturbance changes them, particularly how biofertilizers, alien genotypes change what's there. So I'm going to backtrack from my research program. This is Pinus radiata, somewhere in California where it's native. Because plant communities and fungal communities are so important to each other, it's really hard to establish new plants in new areas without their fungal symbionts, right? Okay, so we know this for hundreds of years. If you go through this Pinus radiata from California, when people were trying to establish plantations, forestry plantations in the Southern Hemisphere, they're taking these North American genotypes. They got the microbes right. So once they got the microbes right, then they could establish these forest plantations, because you either need, when you're establishing a tree that has these specific associations, you need to either use natives that are compatible, you need to use cosmopolitan microbes if they're compatible, or you need to use naturalized invasive microbes. And these microbes weren't there originally when forestry started happening in a commercial scale. However, now they're there, they've been introduced, and when you go to the Southern Hemisphere, you see all these North American trees growing prolifically, perhaps too prolifically. This is a list of wilding pines, right? These are invasive pines that have escaped from forest production in the Northern Hemisphere. This is New Zealand specifically. If you look at this list, you're going to see a lot of the trees we have around here, right? You have ponderosa pine, Doug fir. It's always so funny for me to go, like I was in Brazil last summer, just Doug fir is everywhere in plantation. It's kind of sobering. Anyway, you have to match the target plants with the microbes. We know that. How does this go back to the biofertilizer industry? Well, these are the 3 requirements you need to get alien plants to establish. You have to use native microbes, or use cosmopolitan microbes, or use naturalized invasive microbes. If we're putting those invasive microbes out there already through these products that farmers are using, the microbes are there waiting and ready for invasive plants to establish. This is my fear. And this was, I held a conference in Brazil last year talking exactly about this topic. But unlike that conference, people, at least the commercial people who are interested in the commercial production and the use of biofertilizers, aren't asking the questions that we are interested in. They're asking, are these products viable? They're asking, do they contain what they say they are? Do they contain the right taxa? Do they contain any fertilizer, right? Which a lot of them do. Is there any greenhouse, specifically greenhouse evidence that they work? And the truth, these are all the regulators in Canada require also, right? These are, if you can prove that you're producing what you say you're producing, that it works in a greenhouse, and that they're alive, you can get approval to sell your product, no matter what it is. So it's a very, very loose industry. The questions we should be asking, and the questions I'm asking, is when you put these biofertilizers in the soil, do they persist? Because if they don't persist, they're not really a threat, right? If they die, who cares? If they persist, do they compete with native soil communities? That's the problem. If they don't, if they're just integrated into the community and everything continues happily along, who cares? Do they affect plant communities? Because we know how tightly coupled plant and microbial, soil microbial communities are. And do they affect ecosystems? So no regulation body is asking these questions, and researchers are just starting to ask these questions. Unfortunately, it's about 50 years too late because these products have been on the market for decades. Nevertheless, we still have to ask them. So I'll just give you a couple of little snapshots into my research program. When I was a young researcher just starting at UBCO, I thought it was a good idea to put a biofertilizer into 2 plots on our campus and one on Knox Mountain. So I released the biofertilizer into these natural systems because I was beginning my research program and I didn't understand how, I didn't know the organism well enough. I didn't know what it was capable of. Anyhow, I wouldn't do this anymore, but I did it, and at least we're getting data from it. So here's just 2 snapshots. This was 2013. 2013, I put these isolates in the soil at Knox Mountain and UBC, and I went back to look at it over the years. And these graphs are just the plants' roots that were positive for the biofertilizer. So even at 2022, which is the last time I looked, 25% to 75% of the plants tested positive for the biofertilizer. Okay. But sorry, I keep on going the wrong way. When you look at the abundance, how many were there, right? Remember I said if they're there, low abundance, who cares? When you look at the abundance of them by 2022, or even in 2015, that works out to 1 million spores per liter of soil. And at the very lowest, 80,000 spores per liter. A ton of propagules like that. For reference, I think the product tells you to use 5,000 propagules per unit. Like we're talking, this organism is there and it's there in a big way 10 years later. So they persist. Yes, they persist. Okay, do they compete with native fungi? There have been paper after paper in the last 10 years. Yes, they do compete. They change fungal communities. We know this. Yes, but I want you to hear more about it from Selena Spence, who will be talking about this in detail in a very interesting way that they compete with each other. So I'm not going to steal her thunder. I'll let her talk about it tomorrow. It's tomorrow, right, Selena? Yeah. Okay. What about plant communities and ecosystems? Because that's where I was going from the beginning, right? Do they change plant communities and ecosystems? And I have a grad student right now who's looking at this, and she's just writing it up now. Do these isolates of fungi, commercial and non-commercial, affect plant hosts differently? They do. Whether or not this leads to ecosystem changes, stay tuned. We don't know yet, but this is the focus of my research program right now. Okay, so can biofertilizers invade fungal networks? This is the threat, right? This is the big threat to me, because if they do, remember, biofertilizers are exclusively cosmopolitan taxa. That's what they choose. They choose the ones that are easy to grow, easy to reproduce. Oops. But because they're cosmopolitan and because native conspecifics are present in all these ecosystems that they're being released to, there's a capacity for them to fuse. Remember at the very beginning, I showed you that New York Times article that they grow by fusion? Biofertilizers are capable of fusing with native isolates. And these networks are huge. So if a foreign genotype can fuse with an existing common mycelial network, that is a scary situation in my mind. Okay. And these products are selected for ruderal traits. That's how they pick them. Worst case, I don't know. This is all conjecture. My fear is that this is what a biofertilizer is going to invade an ecosystem. This genotype is going to take over, and we're going to end up with these really low diverse plant systems. I mean, probably not, but we don't know. We don't know what's going to happen. So the take home here is invasions below ground affect invasions above ground. They're linked. Be careful what you add to your soil. I want to thank my group, and yeah, any questions? Yes, Chandra. You need this?
Okay, and I'll just mention that online they asked us to introduce ourselves. So Chandra Moffat, Agriculture Canada. Miranda, I loved your talk and I have a lot of light bulbs going off, but I was wondering if you could sort of speak to biotic resistance. And just one thing that I was thinking about is if we're introducing biofertilizers or other fungal species into ecosystems, how then if those systems may become like the dominant fungi may be the ones that are applied, like how that might affect biotic resistance to invasive fungal pathogens should they be introduced to the system. Is that clear?
I have no idea. I mean, the first question I asked at the beginning of this is the complexity, right? It's the complexity, the interactions, the incalculable number of interactions that these products are going to have with all the players in the ecosystem. I don't know, but then, I guess I get really upset about the regulation issue because we're so far behind knowing what the interactions are going to be like. But that's a really good idea. We should talk.
Yeah. And then just the other thing I wanted to mention is we're not just applying them from a fertilizer perspective, but entomopathogenic fungi that are used in organic agriculture, which is something that my team knows I speak out quite a bit against, is the use of like organic pesticides for insect control, because the impacts of entomopathogenic fungi are also very poorly understood and have a lot of downstream impacts.
It's scary, right? Yeah. Introducing entomopathogens into the environment and just say go, especially what's happening to insects.
Yeah.
That's scary stuff.
I'll talk to you more about that too.
Yeah,
Thank you. Very
totally.
interesting.
Greg?
I may not have understood your graphs, but it looked as though the trend line was downwards.
Oh,
isn't that good?
no, because it's kind of, 10 years later, like I would have thought, because I can't remember when I was doing it every year, and then I took a big gap and I went back to see. They're still there. They're not, and the numbers, yeah, they're down from original, but they're really ecologically important numbers. They're not small players in these systems. Anyone else? Yes. Purple shirt, yeah.
Great talk. Thank you. This is Rebecca Nelson speaking from Utah State University. And I'm really curious whether like you or other folks in this field have looked at how these like biofertilizer based agents, like if they can have like downstream effects on like plant chemistry of like the leaves or the floral rewards in ways that might differ from native fungi, or yeah, how much information is known about that?
I mean, here's the complexity, right? This complexity, the interactions. We do know that fungi do affect plant chemistry. They affect rewards. They affect pollination visits. We know all this. We don't know how these products are changing that. We don't know. Yes.
Spread from a field, let's say they're put on an agricultural field, and we know they're making stores in the soil.
Do you know how far they're going to spread into a natural area nearby?
They spread. I mean, I've done some of this work. They spread quickly within the first season. Within the first growing season, they can spread 10, 20 meters. That's in a disturbed site. They do spread into native systems because there are available hosts there. And we know they travel through the water column. We know that they travel through the atmosphere. So they're microbes. You know, we know from epidemiology, they move really fast, especially if humans are helping them.
Hi, my name is Brandon. I'm a grad student at SFU, but before I was a grad student, I was working for local government as a restoration ecologist, and there was a product, like a mycorrhizal kind of spore powder that you mixed with water product that was marketed to me, like in my role by a company in the States, and I was just wondering if like there's been any pushback against like these biofertilizer companies, or like, you know, this company I'm thinking of that's marketed specifically for restoration. Like, has there been any kind of like government or academic, like kind of like targeted pushback?
Not government. In the last couple of years, there has been just examination of, are we doing the right thing, particularly in restoration? So there's been some work. And now there are companies harvesting natural fungal communities from intact systems and marketing that as mycorrhizal inoculum, which has its whole other set of problems. But there's also good research showing if you can find, you know, remnant target ecosystems, you can culture your own microbial inoculants. And that's, so far, that looks like the best way to go. Yeah.
All right, Dr. Hart, thank you so much for that thought-provoking presentation. Definitely giving me The Last of Us vibes for anyone who's seen that. I'm really looking forward to hearing more from Selena later in the program. So thanks again, Dr. Hart. And if we can have a big round of applause. All right, fantastic. Thank you so much. I am so pleased to introduce you to our next presenter, Mark Affelbacher. Sorry, I did even practice that one. The Chief Operations Officer at CD3 Systems. Mark is a product developer, environmental scientist, and professionally trained landscape architect. Mark has worked for 15 years at the intersection of business innovation and environmental initiatives. He's created products, companies, and programs that increase effectiveness, enhance brands, and improve the quality of lives. Please welcome Mark, who's going to be sharing with us building capacity to prevent AIS in the Columbia River Basin. Thanks so much for being here, Mark.
Thank you for the kind intro. excited to talk with you about this project that we did in Washington State. But first off, I come from the land of 10,000 boats. So I'm coming here from Minnesota, and I developed this technology in part because there's so many watercraft in the state of Minnesota. There's 825,000 registered watercraft. That's an amazing number, varies between that and 850. So 1 in 6 people in the state have a boat. And with 10,000 lakes, 60% of those go to more than one lake. So a lot of this approach and idea that I'm going to talk about in this case study in the Columbia River Basin stem from the work that we started out with in Minnesota and has spread across the continent. Not quite like fungi, but somewhere close. So we'll talk a little bit about the scale of the invasion, some of the boater needs, some of the capacity building that we've done, a little bit more of the gory details on the case study, and then talk about some of the new containment strategies we're doing with check-in, check-out programs and initiatives. So I was researching the context and the cost to aquatic invasive species in Canada, and the best number I could come up with was $5.5 billion a year it costs the Canadian government. And that's only for 16 taxa that they're targeting. That's a lot of money for just those few aquatic species. And right now, across the entire provinces, we know that the current solutions just can't scale. The challenge is getting personnel out in the field. The challenge is getting hot water decon systems. And what we've really researched and found is that the boaters really do end up needing tools to take those actions themselves. And really, this approach grounded itself in economic reality. The land down under, Australia, is the land of invasive species. They've done a lot of research on what costs go into prevention of invasive species and what costs go into the actual management of those invasive species. And this is an invasion curve. The idea is that what is the cost, what are the returns from an economic level on when you invest in invasive species and when are there not returns? So the old adage, an ounce of prevention is worth a pound of cure, isn't quite right. It's actually for every $1 you spend on prevention, it's worth $100 when you're dealing with management of the invasive species at a large scale. So right now, there's some great messaging that happens across this province of clean, drain, dry. That's tied to your local communities and partners with good signage and activations. Well, people plan to act, and what happens is they don't act. And the groundbreaking research that was done initially on this was done by Canadian Council on Invasive Species back in 2018. They researched thousands of voters. They said, "Hey, what do you need? What are the barriers to your actions?" And this is a word cloud of what they found initially in this conversation. Again, about 9 years ago. It's time. It's understanding their watercraft a little bit more and having equipment or a station to clean their boats. This has been done now in Minnesota, Michigan, Utah, and Washington State, and basically the same thing has come across, that the public needs to understand how to utilize clean, drain, dry, best management practices on their own watercraft. And it's kind of amazing to think about that. I get to travel all over the country and go to these wonderful lakes, and every lake is special. This lake is amazing, by the way. It's really beautiful. Probably top 5 I've ever been to in my entire life. But the boaters are the same. You have the same mentality as a boater. And that's a really interesting thing to think about, is that, sure, the lake is unique, the ecology is really important of that, but people think the same across the entire continent. So in the context of the Columbia River Basin and thinking about larger watersheds, do you shield or do you contain? And let me just kind of introduce these two principles, because this is the paradigm that I work in, is shielding aims to protect new invasions. So you're targeting inbound watercraft, a new boat coming into that lake, river, or stream. Or containment, are you trying to hold that invasion at that location? So you target outbound watercraft. It's a completely different approach. So this is where we're trying to identify what strategies you'd like to implement. In this particular project, we're looking at shielding new introductions into the Columbia River Basin. Now, both of these really have to leverage the knowledge set that you all have on understanding what's going on in that water body. So for example, you may have information from inspectors on when people are launching, what kind of watercraft are launching, what's the frequency of use, where are they coming from? All that information can get plugged in to identify, what you're going to do at that individual axis, because when we started this project, this is the number of boat launches we had to look at in the state of Washington. So what the heck do we do? Do we shield or contain? Where are those invasive species at? There's some great databases out there on different boat launches, how big they are, how many people can park at them. It's really interesting information. But again, for this particular approach, we had to work with a lot of different partners. And that partner really represented the best coalition we could get at that time. Now, the federal investment came in the form of national parks. We had investment from some of the other Department of Interior programs, Fish and Wildlife and Bureau of Reclamation. And then we worked with a lot of counties. We worked with Game Fish and Parks. We worked with Washington Parks. And then also we developed and proposed the grant with Wildlife Forever. So our goal was to assess the threat based upon the economic risk, the environmental risk, where it's at in the watershed, what downstream impacts we would have with introduction of dresinids or other invasive species, what kind of interstate or interstate activities we have in those locations, and then ultimately, how can we address some of the staffing costs? Because we don't have enough money to get people out there. We don't have enough people to get out there either. And again, looking at this from the lens of infrastructure is really important because at the end of the day, we should be investing in long-term assets that have a long lifespan. So within this context, we created a hierarchy, understanding, what's the long-term cost-effective solution that we should approach this with? How does that relate to the biological efficacy? Because as you all know, anyone that studies aquatic invasive species, if every single element of a watercraft and trailer could get 140 degree, I'm sorry, what's that in Celsius? Anyway, really hot water for 10 minutes, it'd be fantastic if we could all have them in hot water dip tanks and take a hot bath. Sweet. But those things cost millions of dollars. So how are we going to look at this from a biological efficacy with long-term cost effectiveness? So what we decided to do was self-service equipment with 24/7 availability that had good data on and had a long lifespan. So first off, we looked at some of the different interstate crossings in the state of Washington. Fortunately, many states in the U.S., especially out west, have a border patrol where they check every single watercraft coming in, and they're actually making tickets that have teeth. We're talking like $750. So if you blow by it, you're going to get a big ticket. That's a lot of money to anyone. So they have port of entry locations, and a lot of them look like the above picture. Typically, it's some kind of a pull-up area where they've got some containment underneath that to blow down and address your watercraft. But then when we're looking at that all those different accesses, we try to figure out a hierarchy of which ones are the most important ones. And we use a site evaluation process called access, which is to assess, coordinate, communicate, evaluate, supply, and then standardize the locations. So again, throwing on my landscape architect cap, it's looking at the site, movement of boats, how do we get this normalized into the behavior? How does this go into that targeting inbound watercraft? Again, we're looking to shield, so targeting inbound watercraft is really important, or versus contain. So we use these site evaluations to look at the locations, and we leverage some of the resources that are available. If you do work in aquatic invasive species, there's some really good resources from the state's Organization for Boating Access, SOBA. They have a number of really good resources, and they boil down to something really simple. Tell them what you're going to tell them, tell them what you told them, and then give them space. So let me walk through a little bit on an idealistic site, what that looks like. So first off, you got to have a sign, boat launch here, boat cleaning station this direction as you enter. And then on your way in, stop here to clean your watercraft on the way in or on the way out. This way, again, using line as a directional tools, pavement markers work really great, arrows work really well, because again, people want to follow the line. I don't know if you guys have been at boat launches lately, but I may have seen a couple divorces happen at them. It's crazy. Some people get so angry. So again, then on the way out, we want to give them a message at that location to clean, drain, and dry. So we put in these accesses of free-to-use waterless cleaning tools. They're CE3 systems. Again, we developed these in concert with a lot of different partners to create the best assets about 9 years ago. So for this project, what we have is a waterless system that has on the left-hand side, air blower system. It's push button operated. It has instructions on the front how to do it. Lights that illuminate into and under the watercraft and trailer. In the middle section, we have hand tools that are tethered with a braided stainless steel that you can't cut with a Leatherman. They have 50 feet of self-ratcheting cable and they're plastic coated. Brush, plug wrench, and grabber tool with those. And then on the right side, we've got a wet dry vacuum system. This technology is all solar powered, off grid, has an internet connection through a cellular gateway. So we get data on when and how the equipment is used to inform strategies and approaches. We have customized instructions, and I'll cover this a little bit more on the units themselves for the little vignettes on how to clean 7 different watercraft types. And again, it's important that we cover all the bases here because there's a lot of different boat types out there, but the nooks and crannies of them are pretty similar if you break them down to different watercraft types. And I was able to partner with a couple different app companies, one of which is Fishbrain that does a lot of angling apps. So the signage at the sites look a little bit like this. Again, they've been able to invest in these at some of these strategic locations, telling them what we're going to tell them, tell them what you told them, and then giving them space to do so. So this is one of the units installed. This was at Steamboat Rock on near Lake Roosevelt, if you're familiar with that, on the Columbia River. And we partnered to do a lot of outreach and engagement. I heard something crazy the other day. I read it that kids these days, they look to online influencers for their health information versus doctors. I got a little sad. But online influencers are really helpful in many instances at getting people to get the word out. Having it become popular, have it become normalized, have it become a positive behavior. Those are really good things that can happen through having partnerships that do outreach. So we help with that, with press kits, other resources for your campaigns too. So what to expect at an access when you're there, tools get used. You may have piles of weeds from the proverbial bearded boat coming out. And for every one person that leaves it a complete disarray mess, another person actually does clean it up. It's kind of how it works. The equipment is ruggedized to live outside its entire life, and tools do break, but people have a really hard time breaking them. And fortunately, we've been able to have some cues for care, which are part of making a unit endemic, is making them communicate the local values, whether that's your hook and bullet club, whether that's a watershed group, whether that's a friends of group, getting their logos, their decals on the exterior of them, make who would be a bad actor think twice about taking those actions to vandalize a property. The data is pretty cool. We can get information and maintenance alerts on them, timestamps, auto reports, export it, slice and dice. The person that I work with on this, she has a coffee cup that says there's a spreadsheet for that. So they really went through the data really well. And one of the interesting things we can see is trends by month. That information can be communicated to the grant partners. It can be used as an information tool for newsletters to say, hey, during the week of July 1st, you know, we had over 1,000 tool uses, and here's how the tool uses broke down between our different tool types. It's a great way to communicate the value of it. So the results, we, instead of doing I think it was 850 accesses, we did 55 accesses that we reviewed. We had 20 site improvements, 18 installations. We got about $400,000 a few years ago to do that. We had some match come from a number of grants. We had a number of in-kind contributions from other partners too. And to date, we've had, well, as of last year, we had over 30,000 tool uses. We had over 15,000 sessions of use. So when we're looking at the outcomes of a program like this, we want to point to something, not just the physical equipment, but the real data. And we have smart algorithms to make sure that the numbers aren't juiced, to make sure that that 30,000 actions from the tools is accurate. And I can go into that in more detail, but not now. So future capacity building, again, this equipment isn't inexpensive, and you want to put it at accesses. Thanks to COVID, we can't thank COVID for many things, but one of them is using QR codes. They're common again. People use them. I thought they were going to go the way of the wall telephone, but no, they're actually really useful. So we've created online programs that are web apps that use QR codes to engage the public on best management practices. Again, going through the 7 different watercraft types and not having a boat yourself. The data comes in, you go in through the website, and then you can go through those steps again, and we've got some new partners to take those actions. So looking at this across the entire continent, I know when I work, we've got CD3 systems in 38 states, 4 provinces, and 16 tribal nations now. And I started this by saying I come from the land of 10,000 lakes. Why? Last statistic I heard, Ontario province has 70,000 lakes. It's an amazing thing to think about how many water bodies there are out there, and a lot of work we need to do. And thank you all for doing the work that you're doing on aquatic and terrestrial invasive species. So with that, let me just flick through this because we're running out of time. Conclusion, AIS prevention is key. The top reason voters don't act is lack of tools. Building partnerships is building capacity. It's really important to find those partners that care about your same assets and prioritize your accesses and use your resources to figure out which ones are better than others. Integrate some standards and then execute and educate. So with that, when you think about your messaging for clean, drain, dry, and you plan to act, think of tools, think of resources to take those actions. Because truly across the entire continent, everybody does like a clean boat. Thanks, everyone.
Perfect. Thank you. All right. Well, thank you so much, Mark. That was really great to hear about and learn more about how your research has informed this product. And I love the coupling of the education with providing the tools. I want to open it up for any questions from the audience here. All right.
Do the users of the tool enter any information about who they are and where they're from? And because I'm wondering if you could couple your tool with at least a voluntary information providing that would help people understand the direction of flow of some of these invasive species, or the boats, like our boats always going from, Minnesota to Washington State or anything like that.
Great question. So there's active and passive data you can get on that. Active data is someone puts in their hull ID, which is a common denominator that you'd need to align with a number of the programs that look to identify which water body a boat was at last. So that information you can do with some kind of a check-in, check-out form, and that goes over some of the steps that you have for user privacy issues, especially in Canada. They're very strict. But then there's passive data. Your phones are tracking you right now. There's a catalog of 5 years of that data sitting in a database right now. And we're working on a couple of different partnerships to identify those behaviors, because you can use geofencing to figure out where people are and where they go. It's truly an amazing thing. It's data intensive, using AI to be able to inform that information. So that's something we're working on. We've got some grants that we're proposing to do with the Army Corps of Engineers on that. Great question.
Hi, I was wondering if you could talk a little bit more about how you determine whether the influx or outflux or direction of flow of invasive species is going to be addressed at any certain water access, and whether that differs at different lakes or whether it's usually one versus the other.
Sure. About of all the accesses I've looked at, and I've looked at thousands, about 60% of them, because of the size of them, have your location that you do your clean out is a both location. You do the same spot for the inbound and the outbound, which is fortunate. Now, of that remaining 40%, usually those are accesses that have a design where there's a distinct flow, where people come in and come out, versus more of a, think of it as a linear product or linear location versus like a cloverleaf kind of design. So of those locations, your question was about how do we identify and prioritize those? We try to get the ones that had both, just because we then could get people to normalize their behavior. Because what we found from exit interviews and research, we do a secret shopper research on these things too, where like people act like they're reading a book and then they take notes on what people do. We found that people like CD3 systems or cleaning equipment that's self-service because they're not getting watched, and they'll do it again because it's in their own best interests. So I think I answered your question in that we try to target the both locations. We did some that were just inbound as well. Good question.
Hi, I'm Christina White with Okanagan Nation Water Board. Just wondered, are there final inspections? Like, how do you know that they have effectively eradicated or removed invasives from the watercraft?
I've got a whole other deck on biological efficacy for waterless systems versus water systems, versus hot water systems versus hand removal. So the good news is that the most biologically effective tool for macrophytes is the air blower and the wet dry vacuum. If people take the 3 to 5 minutes that is normally taken on that and they use those two tools, we know that they're anywhere from, I think it was 92 to 98% effective. And most of those species that are going to another water body, now we're really concerned about anglers that are going from lake to lake hopping, because it's about an hour drive people are willing to take on a day's notice. We found that outside of an adult zebra mussel, most of those species, including veligers, die within 2 days, even in some of the best conditions. Research has found that veligers, basically if you sneeze at them, they die. Lack of water, oxygen, temperature change, physical abrasion, lack of food, all those things, just they die because they're spawn feeders. So outside of an adult zebra mussel, and this isn't a tool for that, to be clear, adult zebra mussels are different, and that's a boat that may be in a lake for over 72 hours is kind of where the science baseline tells us. For day boaters, this technology is extremely biologically effective. So again, it's ranking your hierarchy between, are you a day boater or are you a moored boat that's been here for a longer period of time? There's a completely different decon level that you need to do.
All right. Any other questions for Mark? Well, thank you very much. Really appreciate that. All right. Yeah, and Mark, you mentioned the land of lakes, and I think that this is probably the land of lakes in BC. So really topical here. All right, really happy to introduce our next presenter, Stephen Brownlee. Oh wait, are we on the wrong? Oh, gotcha. Okay.
Spoilers.
Wait. Okay, perfect. All right, Stephen, I'll give you your introduction. Thanks for your patience there, and sorry about those spoilers. So our next presenter is Stephen Brownlee. Stephen is a PhD candidate at SFU, working in the Cote and Moore labs. He's a certified indoor ecologist using spatial modeling tools and remote sensing to tackle conservation problems from sea star wasting to zebra mussel invasions. Please join me in welcoming Stephen, presenting Predicting Potential Invasion Pathways and Lake Susceptibility for the Arrival of Zebra Mussels into BC.
All right, well, thank you everyone. Thanks for the introduction. And I'll sort of jump directly into it. We heard a little bit about zebra mussels beforehand, but it's probably worth getting into the actual biology and ecology of these mussels, because I'm sure you've seen all the signs and heard about the campaigns in the news, but you may not know all that much about them specifically. So zebra mussels and their closely related species, quagga mussels, are mussels that we believe are originally from the Black Sea and Caspian Sea in Europe. We don't know exactly where. They're also a rare example of a species that was named after another species that actually does seem to fit the name. You can see the stripes kind of align with one another in both of those examples. One's named after a zebra, one after a quagga, which is a related species. So it's sort of nice that way. It's nice that it's finally an example of a species name that actually fits. So the unique thing about them, as far as freshwater mussels go, is that they have a number of characteristics only shared by marine mussels, like planktonic larvae called veligers, like we heard about, and the ability to produce sticky byssal threads that allow them to attach to hard surfaces, just like marine mussels do. These traits also make them proficient at dispersing using human transport systems, as they can attach to hard surfaces, as we found out, and be transported in the ballast systems of boats or in the sort of water that collects in the bottom of boats in veliger form and be transported very long distances. This is exactly how they got to North America, in fact. So they were transported across the Atlantic in ballast water and carried all the way to the Great Lakes, where they quickly established themselves and began dispersing further through overland movement of boats, hopscotching eastwards and southwards downstream and spreading further across the continent. The depressing part of when I have to give these talks is that every time I make a range map for them, I have to update it with more locations. Like this one in Idaho for quagga mussels, which is by far the closest location to us. Their arrival here is obviously a very imminent threat, and it poses major risks to freshwater ecosystems across BC. Mussel-infested lakes in their invaded range experience major crashes in native freshwater mussel populations. And while we don't know what their impacts on salmon would be, we can assume that it wouldn't be good. Understanding the processes by which these mussels can get transported here and which lake systems are susceptible to them are super important to preventing this imminent invasion from actually occurring. And that's what we're hoping to accomplish with this project, which tackles both halves of the process of invasion, specifically a network model to examine the routes by which zebra mussels might theoretically arrive, and a suitability model to predict whether or not mussels might be able to establish themselves in a given lake. So we'll start with the suitability model and move outward from there. So previous mussel suitability assessments used a small number of hand-picked environmental variables and relatively coarse environmental data sets to produce assessments of suitability. So you can see an example here where they used the sort of underlying geology of a given region to assess the calcium concentration present in a given region and therefore its suitability for the mussels. We're hoping to improve on that, like we said. We conducted a literature review to identify variables that might contribute to mussel success or failure in establishing a given location. And we identified carbon concentration, salinity, calcium concentration, and temperature as key drivers of mussel invasion success or failure. We've also been able to take advantage of the rise of remotely sensed imagery to peer directly into lakes from space and measure the variables we're interested in at very fine temporal and spatial scales. So specifically, we're able to collect data on 3 of our 4 variables that the literature review identified. Salinity was not particularly relevant for an inland lake perspective, but so we discarded it in this case. We have surface temperature, which is directly measured by the satellite. We have a fancy looking index that basically measures the spectral signature of calcium carbonate in the water directly. And we have chlorophyll A. These were all collected from the European Space Agency's CCI Lakes dataset and used to assemble a 2-kilometer square resolution dataset for a collection of lakes in BC. For the purposes of today, we'll be looking at Okanagan Lake specifically, since we're here and it's a major potential front in the evasion of these mussels. So here's an example of what the environmental data we're looking with looks like. In this case, we've aggregated it into a month interval, specifically July 2020, in order to fill gaps caused by cloud cover. Now we have more or less complete coverage of the lake and a time series going back all the way to 2002. You can see again the projected, either measured directly or predicted values for all of our variables of interest. So the next step is to pair this environmental data with the physiological data we need. Specifically, we need to assess what impacts the environmental conditions present in a given lake will have on mussels' physiology if they are introduced into a system, which is the sort of baseline mechanism of how invasions succeed or fail. So with our environmental data in hand, we went back to the literature to identify some key physiological limits the mussels experienced for each of our environmental variables. So with temperature, their tolerances were bounded between 0, which is the freezing point of water, which makes sense, and around 30 degrees, depending on a variety of factors from acclimatization to the source population. At the southern end of the mussels range, they are much more acclimatized to hot temperatures than, say, one from Minnesota would be. But in our case, we've decided to set the boundary at 0 and 30 degrees. So calcium concentration, like I said, is a vital limiting factor for the mussels because they need a sufficient amount of calcium in the water column to be able to build and maintain their shells. If it's not present, they'll wither away and they will fail to be able to reproduce themselves. As you can see from this data on survivorship of mussels at different calcium concentrations, there's a pretty strong inflection point at about 28 milligrams per liter. But there is that one lone annoying population that is able to survive very well at much lower calcium hardnesses. These are extremely hardy mussels, and they are frustratingly difficult to actually find out what can successfully kill them. So in our case, we have set the lower bound for the mussel survivorship to be about 10 milligrams of calcium per liter. Again, under the assumption that there are populations out there that could theoretically be donating mussels to a given system that have these tolerances, either genetically or acquired. And then finally, mussels as filter feeders need a sufficient concentration of organic particles suspended in the water column to be able to filter feed, and which experimental trials have identified as being about 0.36 milligrams per liter of particulate organic carbon. So you can see the horizontal line and the vertical line that the authors identified as being the sort of break-even point for the mussels. We can use a conversion ratio developed for oceanography to estimate the particulate organic carbon present in the lake as based upon the concentration of chlorophyll A in the water column. In this case, chlorophyll A is a very distinct spectral characteristic, so it's easy to detect from satellite imagery. With all this physiological information assembled, we can use it to construct an assessment of suitability. So in order for a given set of conditions to be deemed suitable, all 3 criteria need to be met. So if any of them fall outside of the bounds of one of those 3 steps that we've laid out, the conditions are deemed unsuitable. Each point in time and space in our great big satellite data set was assessed against this set of conditions and given a sort of a simple binary unsuitable or suitable assessment. So the results are pretty striking and show a pronounced seasonal pattern of suitability. So I've sort of plucked out a demonstration data set here. Here's a month-by-month suitability assessment for Okanagan Lake from October 2019 to April 2020. So you can see as the lake cools down over the fall and winter, it becomes dramatically more unsuitable for the mussels, and this recovers only in the spring of next year with sort of defined growing season in July and carrying on through October. Again, the cycle repeats itself. So the question sort of begs itself, how does suitability change over time in this lake system? The answer is that in Okanagan Lake, at least, conditions are too cold and low productivity. Those are the 2 factors that tend to fail in the sort of a suitability assessment framework. It's mostly cold and low food levels than to be able to support mussel growth. Summer season and early fall are windows in which the mussels can grow and theoretically reproduce, since large chunks of the lake are suitable for them. And maybe more importantly, shallow, more productive areas of the lake remain completely suitable all year round, which is important because those are the parts of the lake where the mussels are likely to be introduced, since that's where you put in your boats that might be coming in from other locations. So the next step in our sort of 2-part assessment is we know what the lake's susceptibility to mussel establishment might be in terms of their physiological characteristics and the conditions in the lake. But the question still remains that we need to answer how they might arrive and where they might arrive. So to do that, we're going to use a network model to describe all of the possible routes a person transporting a boat overland might take to get to the Okanagan and assess them for their risk of exposure to the mussels. So we'll start with a collection of cities, specifically every city or county in the US or Canada with a population greater than 20,000 people. So from there, we use a route finding algorithm, which happens to be the same one Google Maps uses, which is nicely circular because it's the algorithm people will be using to find a route, and also a efficient route finding algorithm in and of itself to plot a path from a sample of locations within each county or municipality in Canada or North America to the Okanagan. You can see here what it looks like if you lay all of the individual routes on top of one another and then color the resultant raster by density of routes. It has this very nice, pleasing dendritic shape that sort of mirrors the actual river systems in North America in a nice way. So analytically speaking, the next step is that we take each of those routes and split them into a long chain of 2-kilometer long segments that can be laid out in a chain, like so. Each segment is then assessed against its distance from a zebra mussel infestation. In this case, specifically against this distance surface generated from a database of occurrences. Logic behind this being that the farther away a given point along a traveler's route from an infested water body, the less likely they are to be exposed to it through diversions or recreation. So using this information, we can set a threshold for distances that we consider to be within an envelope of sufficient risk, say an hour's drive from the route, and then calculate a collection of other risk metrics which capture information about the configuration of the route and when the mussel exposures occur. So exposures along the route capture, in this case, we have exposures along the route, which captures the total number of exposure events along the entire route. Buffer distance at the point of origin, which tells us about the conditions present at the point of origin, whether or not it's likely the boat is quote unquote naturally in an environment infested with mussels. And then finally, the distance between the last exposure and the destination, which attempts to measure the theoretical shortest distance between an exposure event and the Okanagan, which is important to determine whether or not the mussels will be able to survive that long distance. So here's a selection of representative lakes to show you what this looks like in practice. You can see that the number and distribution of potential exposure events varies significantly across the routes. And if we look at all the routes collectively, a pattern emerges. You can see that exposure events, as an example, you can see there's a very strong east-west structure, and the further east you travel from, the more likely it is that you're exposed to mussels at some point along the way. And you can see a very similar structure if you look at our other metrics as well. So large-scale take-home messages. So Okanagan Lake is at a very high-risk mussel establishment, despite low productivity and cold temperatures in the winter, and there are high-risk areas in the shallow areas of the lake that are suitable year-round. You might also experience some similar dynamics where there are die-offs in unsuitable areas and recolonizations, but we don't really know enough to say for sure right now. And it highlights the importance of intercepting mussels before they arrive. Route modeling allows us to extensively plot out the paths by which the mussels might arrive and determine that further east the given traveler hails from, the more likely it is that they are exposed to mussels. And that's about it. Thank you.
Maybe just hang out.
Hold on, yeah.
Fantastic. All right, Stephen, that was an awesome presentation. Thank you so much. Really illustrated, I think, the importance of the indoor ecologist. So thanks again, Stephen. Before we dive into questions, I know Chandra already mentioned this, but we have had a request to just say your name and affiliation before your question, just to help out the folks online. So with that in mind, love to open it up to any questions for Stephen. Oh, we've got one at the back there, just with the mic. Yeah. Hand up if you, yes. Awesome.
Christina, Invasive Water Board.
Is anybody looking, like, I believe there's golden mussels now and they have a lower tolerance. Is anybody looking at this in a similar way?
Not that I'm aware of. As far as I can tell, there's a lot less research being done on golden mussels, and especially about their sort of fundamental physiological constraints. I would suspect it's probably too cold from them here, just based upon the fact that we're sort of at the very northern edge of the zebra mussels range as well. But yeah, as far as I know, no one's done it yet. They're a very new invader here at least, so it's a good opportunity for me to get on the ground floor and expand at some point in the future.
Absolutely. Thank you.
Sandra Gillespie from the University of the Fraser Valley. I was thinking about native freshwater mussels, and I was wondering if anyone's looked at the tolerances and locations that are most suitable for the native species, because you might have a strong overlap between the places where the invasive is most likely. They need the same things. So you might have locations for high risk of impacts on natives as well. And I was wondering if there were differences and refuges, and sort of if you had any insight on any of that.
You've anticipated my third chapter, which is not really. It's always great when that happens. But yeah, there's absolutely a lot of overlap in the habitat requirements. They need hard substrate as well in order to attach the zebra mussels to attach to rocks as well. In the native mussels case, they like to embed between rocks and different substrates and use the sort of flow around them for feeding purposes. So I know in Ontario, for example, where both species of mussels have been for a long time, they have the zebra mussels and quagga mussels will choke out unionid mussels, the native mussels, and just infest them entirely and cover their shells when they are actually exposed. So I would expect, at least here, the impact will be very severe.
Hey, this is Brandon again from SFU. And I was wondering, you mentioned that one population that can tolerate lower levels of calcium carbonate. I was wondering, do you think that's evidence of these mussels being able to like evolve and become more tolerant over time? Or do you think that population could have had a different like source, genetic source?
So this is from a paper by Mackey in 2010, I believe, and it was sort of a natural experiment they did in a series of lakes in the the Canadian Shield in Ontario, and I believe that lake is sort of a naturally very acidic lake. So the assumption that the authors had, they haven't done genetic testing, but is that they were introduced to that system and sort of adapted within that lake system to the lower pH and lower calcium concentration conditions that were there already. So I think it's absolutely a case of them adapting to new novel conditions.
All right, we've got one more question over here.
Hi, Stephen. This is Jofina from Fisheries and Oceans Canada. I just had a question with the surface temperature data that you have. During your research, did you ever have identify a need to collect temperature data from like the bottom of the lake or, especially for stratified lakes? I know it's a lot of data collection if you were to do that, but just curious if you had thoughts on that.
Definitely a need. That was one of the things we ran into very early on, is that as a naive grad student, I was like, oh, surely there's lots of epilimnion and hypolimnion temperature data out there I can use to model this. And it's obviously very time consuming to collect and to deploy on the scale that you need to match some of these remotely sensed data sets. So there's absolutely a need. And that is one of the limitations here, is that we are measuring surface temperature. So it's more extreme in terms of summer and winter, for example, because it's the most exposed to the air and therefore hottest and coldest. So.
All right. Thank you so much, Stephen. Really appreciated that great presentation. Maybe two. Yeah, fantastic. Thank scientist at the Pacific Forestry Centre in Victoria, BC, with the Canadian Forest Service, studying evolutionary genomics of invasive insects. Please join me in welcoming Gwilym, who is joining us virtually and presenting collaborative tool production for invasive species management, a review, and 2 case studies in genetics.
Hi, just taking care of a little setup here. Okay, that should work. Thank you.
That looks good. You can see this in these slides moving.
All right, well, thank you, conference organizers and participants, especially to the organizers for rolling with the times and keeping this running smoothly. My name is Gwilym Blackburn. Advancing my slide here. With a spacebar touch, I thought. So yeah, what I'm seeing are grayed out buttons. Is there a way to grant me control of my slides?
Hey, Willem, I just approved the control, so you should be able to move it. If you open up the meeting, you should be able to control the master slide.
Okay, not so far. So leave the meeting and re-enter, or?
Try clicking the next slide button within the meeting, and it should let you do it.
Yeah, you're right. There we go. All right, so these are the conversation threads. I'll move pretty swiftly, basically touching on the ideas that collaboration affects tool development and trying to present you a couple case studies that we're working on within our group of how that affects genetic tool development. So no big news to the group here. Invasion is increasing across the tree of life with no predicted decline in the speed of that if left unchecked. And of course, the costs are widespread, whether you look at it across countries, sectors, or individual species. And so there's a big move on, of course, to address that. And collaborative science is widely recognized as part of that process. My own research program covers basically the image you see here. The most important text is in red. And basically, the protocol count on the left just gives an account of the roadmap on the right about how you can connect genetic tools to formalize knowledge and how that knowledge addresses everyday here and now questions of interest to a forest manager or someone managing invasive species. Of course, the roadmap only touches on a small subset of tools available, and the linkages between those and the knowledge they provide are also here shown only in subsets. So really, it's a very creative field where a whole array of different methods are being brought to answer useful questions. For that reason, genomics pairs very well with the traditional ecologically based management that we've been using up until the 21st century. Literature up until the present widely cites the gap that is recognized between genetic and other sorts of tools put out by research and management, either voiced as prospects for genetics to enter management domain, or management voicing issues and the needs for tools that can be clearly provided, at least in part, by genomics. And so what I feel in the message along this thread today is that collaborative science has a part in sort of bridging this gap and helping genetics earn its place in modern forest and invasive species management. My first pass at directly addressing this or studying this was with a collaborative group out of a research project that I worked on during my postdoc, in which we developed what we call an applied empirical framework aimed at designing tools that are needed and getting them to the point of production and uptake at the other end by the use of collaboration. Of course, a tool in order for it to be adopted and used needs to have a soundly biologically founded design. But as we argue in this review, its acceptance and its conception is well served by a broadly collaborative effort in which you appeal to broad practical relevance of the tool you're making and make it accessible enough that it's going to be adopted and used. So just touching on a few highlights that we covered in the review, of course, that review depended on a wide literature in this direction that I encourage you to read. Just showing a couple of the images from our own study. Collaborative science, as you well know, is on the rise. That's very healthy. And it's in part just by virtue of the increased globalization that leads to the invasion problem in the first place due to human transportation and trade. And so what we see here is basically a breakdown in cultural and technical barriers, allowing institutions from around the globe to collaborate more effectively. This is an example with invasion science focused directly on insects. Our study uncovers a few highlights about collaborative science that we find of interest. As we argue, there are various kinds of collaborator in terms of the sources they represent and the institutions that they come from. And they play a variety of roles in a cycle of developing some kind of tool that you'd like to use for management, from identifying a problem and a potential solution to planning through development and ultimate rollout and implementation. The table need not be read in any great detail here, but one thing that we found interesting to explore was the collaborator types that you saw in the previous image can each play a variety of roles. Of interest and direct relevance to the collaborative process is that they each can play a stakeholder role due to their different interests in a given tool, whether it be basic knowledge or an actual street-ready tool. They also have different roles by virtue of the politics that they're involved with, the finances they have, any security interests. They have unique roles as suppliers or facilitators in the collaborative process. And an interesting thing that emerges from that is that for any given tool, whether it be a data system, or a technical instrument, for example, to be developed successfully and ultimately used at the production end, it benefits to have as broad collaborator involvement as possible in order to conceive of a tool that is actually wanted, and it is going to be vetted from the earliest phase of its design and development. That said, choosing a collaborative team is a bit of an art, given that the motives that a given stakeholder, that a given collaborator type brings to a project is also paired with its strengths and limitations as a collaborator. Those strengths and limitations can stem from things like the financing structure it operates under, political limitations from an industrial perspective, for example, patents that might affect a final product. And as a result, the motives, even if they're shared across a given array of stakeholders that can collaborate on a project, does not necessarily equal uptake. It depends on the various strengths and limits of those who participate. So choosing judiciously a collaborative team to build a tool is well advised. And then a finding separate from our own work that I found really compelling and interesting is there's a general trend, whether you look at articles, patents, or software, showing that collaborator group size, irrespective of the composition of that group, seems to affect the nature of the tool made. On the one hand, the rollout of the tool and its promotion and its ultimate success in uptake seems to correlate directly with group size. On the other hand, innovation generally is greater for smaller groups. And the take-home message that we take from that is that large projects that are able to assemble the resources to create a useful tool pair well with substructuring within that collaborative group to maintain innovation and creativity and design so that you're not too burdened and locked into given paradigms, and you're allowed to seek solutions that might be better tailor fit to the product's, the tool's ultimate goal within your project. So from that, I'll leap into a couple of case studies. Both of them exemplify sort of large-scale collaborations. Both are Genome Canada projects. Genome Canada is a sort of mid to long-term funding system that many of you are already aware of. That is a Canada-wide option for institutions to collaborate and generate tools that require a lot of resources, while nonetheless allowing for the kind of substructuring that I was mentioning that allows the tool to be dynamic and creative and to sort of roll with the discovery and development process as you build a given tool. The first of the two studies focuses on a tool that we've been designing in our group that's meant to profile female flight capability in spongy moths. I'll give you a little background for this. This sort of conceptual motive stems from the idea of invasive traits. If you picture an invasive process that's nicely illustrated here from this paper by Roy et al., from transport to ultimate spread and establishment in a new invasive range. For many species, there are particular traits that mediate more than other traits the invasion success of that species. These tend not to be general traits that you can sort of use to apply to all species, but in any given species or species group, there are often group or species-specific traits that facilitate invasion. And so the spongy moth example that I'll give focuses on the latter of these at the dispersal phase once you arrived in a habitat. Here we see the global range of the spongy moth complex, which is comprised of several species, one of which is established since its introduction in North America about 150 years ago near the city of Boston. Of course, the spongy moth is a notorious invasive species. Members of the species complex that I'm discussing here can attack a wide array of tree species and plant species, both broadleaf and conifer. During population outbreaks, and in particular during successive outbreak years, they can inflict wide-scale damage to the hosts that they attack, ultimately leading to decline and mortality. And an interesting aspect that is the handhold for this present study is that although the global range I've colored here in terms of the approximate female flight capacity of this species complex, it's not, in fact, when you take a closer look, precisely representing any given population's flight capability. Males from all the species in this complex can fly and disperse strongly, whereas females of the European form, at least, are relatively weak in that capacity, showing limited or no flight capability. And yet that's just a generalization across the global range. If you actually look at the few locations shown here in terms of colored dots, for which we have direct flight capability data, you can see that there's variation within any given population studied, usually, with a proportion of moths showing flight capability and a proportion of moths showing little or no flight capability. As a result, the question marks you see scattered throughout the current global range or areas that regularly receive incursions of this moth, it's never too clear when you have in the hand a given intercepted specimen, particularly since mostly those occur in the form of egg masses or battered males, whether or not you're receiving a propagule that shows strong female flight capability. And so I've shown in the left here the benefit that you could have of having a genomic tool that could quickly profile a sample in the hand, irrespective of the life stage or the taxonomic identity of that specimen, in terms of profiling it rapidly for flight and therefore dispersal potential, in terms of managing one among an array of populations that you have limited resources to address. That could be used, for example, for the question marks in blue for looking at the variable flight propensity of propagules arriving on different ships, or cargo. Also looking at different invaded sites that are already established in North America to ask, how should we be managing different origins or different incursions that we've detected? And I've cartooned, for example, in the BC area, an example of an annual set of incursions detected within the province. You could also explore the native range and use a tool to profile potentially, for example, new trade routes to assess potential invasion risk by adopting those routes.
Sorry to interrupt. This is your 2-minute warning.
Okay, thank you. And so in short, we're aiming to make a genomic tool that's sort of cartooned on the right here, where you could profile any given specimen for a number of traits. And in yellow, that's meant to represent the one that we're focused on here, which is flight. It's a tool that brings with it little upfront cost and an endless benefit in the years to follow once the tool is made. So the long story short is here, a small part of the BioSafe team that was the Genome Canada project making the tool collaborated across government and academia. Because of the size of the team and the USDA facility represented by Melody Keena on the team in particular, we were able to go down and use colonies that she has in her possession, shown by the dots here, and bring a variety of genomic analyses to that. I was in charge of one of those analyses and then shoring up all the results and bringing it to production. The result was a genetic tool that we can successfully profile at about 88% intercepted samples. And we are going on to extend and corroborate the set of candidates that you just saw with an upcoming study which benefits from a global sample. This is touching again on the highly collaborative nature of the project. The sample was amassed by Sandrine Peak and Michelle Pousson on our team. We'll be using flight tests performed on the one color dot you see on the global map here, together with that global sample to extend and strengthen our tool, as well as to add a geographic component to that tool. And here's just a small sample of the institutions participating in gaining that sample, the global sample for us. The second one I'll touch on only quickly. It's a second tool, this time touching on pinewood nematode, which is an endemic North American species, but poses an invasive threat elsewhere. We're capitalizing on the capacity to use eDNA, which is to sample tissue from any environmental context, in this case Canadian wood products, and test them following phytosanitary treatment to ensure that they don't harbor living pinewood nematodes. eDNA has been quite a recent revolution within genomics, which is itself a young field, and it allows us the ability to sample rare or cryptic tags relatively cheaply and safely. It's a very appealing new tool in development. Cartooned here, because what I want to show you is particularly shown in red. We're interested here in capturing not only eDNA floating around in the wood matrix in which the PWN live following phytosanitary treatment. Once they're killed, we also want to sample eRNA, which represent living transcripts that would symbolize the presence of living individuals. And after treatment, of course, we're hoping to find no eRNA molecules. Again, we have a big team collaborating in the project, shown here in terms of leaders, investigators, and collaborators. A much smaller subset of that team shown here, capturing government and academia as well as industry, went toward making our eDNA tool, as well as First Nation groups across the country participating in the project. Part of the role of the project was to create best practices for eDNA and eRNA tool development and usage, because of course our tool has the capability of testing our wood products for PWN, but of course our trade partners can also test our products for it. So there's a bit of a double-edged sword aspect to the tool. What we're hoping to do is produce a tool in the inevitable eDNA revolution that'll lead to widespread use of these tools and promote best practices for its use. We've done that here in the form of a few guides for practices that I'll show you at the end. The PFC, the center at which I work, the Pacific Forestry Center, contributed to the project in terms of the water bath you see here, which allowed us to do a 56°30 standard wood treatment temperature regime to test our tool. We also upscaled that to testing the samples within real wood matrix in standardized industrial wood kilns, showing the final result here, showing that our tool does work in terms of eDNA and eRNA. We were able to detect strongly the presence of pinewood nematode in our product, and it was able to do so while excluding the presence of congeners, shown to the right on this graph, as well as samples from further afield aspects of the tree of life, showing that our tool is species specific. Other results showed that we are able to test our wood and confirm the presence of eDNA, but the absence of eRNA, showing that our phytosanitary treatments worked. And then finally, as I mentioned earlier, during the course of the project, our team has developed a couple of guides toward best practices, as well as a forthcoming guide on study design and field methodology. And with that, I thank you for your attention and welcome any questions.
Fantastic presentation. Thank you, Gwilym. Big round of applause for you here in the room. Any questions? All right. Gwilym, I don't see any hands up. I'm going to assume that that's because people are hungry and you did such a thorough job of sharing your amazing work even in this short time frame. So thank you so much for being here with us. We really appreciate you. Let's give Gwilym one last round of applause. Thank you. All right, folks. I just want to thank Dr. Hart, Mark, Stephen, and Gwilym for sharing their insights on prevention in our first session of the conference. I think that was a great way to kick things off. I'd also like to just acknowledge our amazing sponsors, including Soil Tracker Solutions. They are supporting our lunch today. So thank you so much to Soil Trackers for your support and for being here with us at the conference. Please go check out their booth when you have a chance. Awesome. Thanks, Anu. Okay, and lunch is actually going to be served just across the courtyard in the front of the building here. So just exit the same way that you came in, walk directly across the green space out front, and it's in the sunroom, big glass area. There will be lots of signage, so head out that way. We're also going to just adjust our start time to 1:15. It's a little bit of a shorter lunch just to bring us back on track. And that's adjusted for running a little bit behind today as well. So let's head out and have lunch. If you've made a special dietary request, please take a look at the meals set aside for you. Thanks again, everyone, and great way to start the conference.
Hi, everyone. Just a message for folks online. We're just slowly funneling back into the room here. So please, thanks for your patience.
Yep.
Yeah. The microphone is Eber.
Oh, yeah.
And the speaker is Tresson. Yep, sweet. Camera. Yeah, it's great.
All right, welcome back, everyone. We are going to get into our monitoring and surveillance session this afternoon. And it looks like the weather gods are smiling on us for a nice field trip after that. So yeah, without further ado, I will introduce our first speaker for the session today. Our first presenter is Nicole Jeans Williams. Nicole is a technical and scientific advisor for Synergy Semiochemicals Corporation. Nicole provides hands-on expertise to support Synergy's valued customers. She's been a forest health consultant in South Central PC for the past 25 years, bringing her years of field experience using semiochemicals to manage and monitor for bark and wood-boring beetle populations in the field. Yeah, so I would encourage everyone to give Nicole a warm welcome to present on optimizing the release device technology for a multi-pheromone blend, notes on chemistry and field trapping of longhorn beetles in BC. Yeah, welcome, Nicole. Oh, is that you or me? You. Okay, up is forward. Okay, thank you.
You guys, can everyone hear me okay? Perfect. Welcome back after lunch. Hope everyone's enjoyed their lunch. Today I'm going to be talking about, like my introduction said, optimizing the release rate release device for a 10 compound blend. Notes on field trapping of longhorn beetles in BC. And this is a talk that was given to the 33rd USDA Interagency Research Forum on Invasive Species, along with our collaborator, Elaine Roque, who sort of talked more about the biology and the trapping of what species of concern we were interested in trapping. But so that part of the biology end of things will kind of be missing, but this is more of the chemistry and development of a release device, trapping device. So invasive species move through international ports of entry, and we are interested in them because they pose a potential risk to our industry. Pushed the wrong way. Oh, going the wrong direction. So this is sort of what Synergy Semiochemicals does. We're a synthetic chemistry lab, and we formulate insect pheromones and pheromones. We formulate, manufacture, and package attractants and repellent devices, mainly for tree pests, including invasive species. We manufacture the traps, which you can see out in the lobby. A lot of us on the team are forest entomologists. We can provide expert and sound advice. We look for research collaborations for new improved lure development for our traps. We produce standard product as well as custom devices for potentially new invasive species. And we're a direct seller, and we have a global presence. So we collaborate with researchers worldwide. Just a little bit about the release device science. We produce controlled release devices to attract or repel insects. We achieve device longevity and consistent release rates with our bubbles and our pouches, and we tailor to the device type and load to its chemical properties. And this is just an example of our verbenone released device. And what we want to see over time is a consistent release of chemical over time, so that we have a really good idea of what we're releasing and how long it's going to last in the field. So a little bit of background. Elaine, our collaborator in Ray, has identified, along with other researchers, a mixture of chemicals that would be attractive as pheromones to a diverse group of wood-boring insect pests, including what would be invaders, to a particular area. This is a mixture of 10 compounds, and this blend, traditionally, when you start out, use a sort of a rudimentary release device in a trap lure. And unfortunately, this means that the device does not last very long in the field. So this is a group of chemicals that we were looking at. And this is the release device that he was using in traps to begin with. Unfortunately, like I said, the cotton wick is not a very well-controlled device. It has a really short field life, which makes it not very operationally realistic to be using at ports of entry, just because someone would have to be monitoring it and changing it every 2 or 3 weeks. The blends of the chemicals in that release device have different release rates. And so it could mean that at one time the lure is attractive to a certain group of insects, and at another time it might not be. So our objectives in 2023 and 2024 were to analyze the chemistry of the mixture. I think I sorry. How do I go back? Because I think I skipped. Yeah, there we go. Can we avoid the creation of the secondary products, which could be potentially a problem? Are the chemical structures of the compounds, are they compatible with the device plastic that's typically used? And do the ratios of these compounds change over time? And essentially, we want to develop an optimized release device for these blends of compounds, assess their lifespan under lab conditions and field conditions, assess the ratios of compounds remaining using gas chromatography, and assess the devices in the field for their trap catch in BC. And we also had collaborators doing this similar research in the States and in Europe. So our lab methods in 2023 were to assess the release rates and the ratios of the compounds in the mixtures in a controlled setting on this cotton wick device. And they were measured gravimetrically in our lure, what we call a lure aging chamber. There were 3 release devices per sample, 12 samples, constant temperature of 30, and weights were taken on a regular basis. And the release ratios of the 10 compounds were analyzed via GC. So what we see is this release rate change in composition over time. This graph shows both a set of on the x-axis concentration and the x-axis is time in days, and the y-axis both concentration and release rate. The release rate behavior show it really after 14 days. We're getting very minimal release rate, and the components really differ in the release over time. So our results show that the cotton wick devices, the release ratios between devices are significantly different. On average, they do deplete after 14 days at 30 degrees C, and sometimes some compounds are gone within those 8 days. But we did have really good success with field trapping with these devices. We did capture a really good diversity of families and genera of insects. 25 families in our study plot in Burns Bog with just 2 traps, and 27 families in Salmon Arm, where I am, with 3 traps. Although the beta traps did catch relatively no numbers of each species compared to more focused lures that we do sell for BC. And that was to be expected. So in 2024, we tried 3 different things. Is the 10 components in a group of 3 different bubbles. And they were grouped based on the boiling points of the chemicals. We did put all 10 compounds in a single device. And then we have a few different groups where we tried 6 or 8 of the compounds. And these are ones that we're now selling commercially. In terms of release rate and ratios, the 3 bubble caps under lab conditions, we show that over time they are releasing quite steadily, and the proportions of the chemicals are releasing well over time. And the release rates are sort of where we want them to be as a target. So our new device characteristics and concerns. So we've demonstrated in the laboratory that the pouches and the bubble caps have a very stable and steady release rate. The blends may result in a change of chemistry over time within each device. And that was showing by the impurities that show up after these have been in the field for a while. So this is for the all-in-one bubble cap. As with the previous experiment with the bubbles, when all 10 compounds are presented, there's a homogeneous and proportional pattern of ratios of each compound that lasts in the entire 120 days. And you can see here noted in gray the sum of the impurities that do accumulate. However, this longer lived device means more time for these impurities to form. Based on these results, we did remove one reactive that was really reactive ingredient from this blend, and we put it in a single device, and that's the epoxynonenal. And in this graph, we see the release ratios of the blends and the same rates of the blends of the same bottle, starting with a release rate of about 7 milligrams per day, decreasing really fast in the first 30 days, and then a really steady release rate of about 2 milligrams per day. As a comparison of the release rates among the 3 bubbles, caps containing some of the same compounds, the line in gray, yellow, and orange, against the one containing all 10 compounds, which is the blue. All of these present different starting release rates, but at day 120, we achieve about 1 to 2 milligrams per day, which is sort of what we hope to target. This is a similar graph, but for the pouch device, and it starts at a much higher release rate, about 25 milligrams per day, and then the bubble cap, and then steadily decreases to about 5 milligrams per day. So what do we learn? The ratios of the compounds are relatively stable over time in these types of devices. The ratios and release rates between field data and lab data are comparable at 25C. The bubbles release rate is about 3 to 3.5 milligrams per day in the field, and in the lab it's very similar. Some blends generate a lot of secondary compounds or impurities, so that we know the reactive and interactive compounds of this blend must be separated from the rest of the compounds. So we have a separate lure that we would use. For the field trapping, we tested in 2024 the 3 different release devices, the bubble, the half pouch, and the cotton wick, in a randomized block design with 15 traps, using the Synergy multi-trap with no fluon. Fluon is often used on a trap just to make it more slippery. We decided to opt out of using that just for this trapping. We trapped June till August, and traps were collected every 2 weeks. And the cotton wick, of course, we had to change every 2 weeks. And the bubbles and pouches were left out for the entire season because they were able to last that entire season. And this is just an example of the diversity that we caught in these traps. A total of 60 species from 25 families. And we did compare the bubbles, pouches, and cotton wicks just to see if they were different in sort of what they could catch. we did see some subtle differences in the species that they could or couldn't catch, but in general, they were all of the devices were really really good at catching that capturing that biodiversity that we would want from a surveillance lure. So our conclusions: the pouches, the bubble, and the cotton wicks attract similar species diversity. The cotton wick, however, must be changed every 10 days for it to remain attractive. The pouches and bubbles last longer, deliver a stable release rate over time, and keep component ratios during the 100 days. The pouch is operationally and economically the better device, and the reactive and interactive epoxy nonenal must be separated from this blend and used as a separate lure. Oh, this is just an acknowledgement of our partners and how you could get a hold of us. And yeah, thanks everyone for listening.
Cool.
All right, and no questions online there, Kim? Okay, all righty. Yeah, thank you very much, Nicole. All right, our next presenter is Jocelyn Haywood. We're getting into the marine realm where I'm most comfortable. Jocelyn is an undergraduate student in the Department of Biological Sciences at Simon Fraser, studying ecology and conservation. She's currently undertaking her honors research on changes in density and distribution of Japanese mud snails in southwestern BC under the supervision of Dr. Isabel Cote. So yeah, please welcome Jocelyn presenting on the influence of burrowing behavior on population density estimates of the invasive Japanese mud snail, Batellaria atramentaria. All righty. Did I butcher that?
This is forward, this is back. Awesome. Thank you for that great introduction. I'm Jocelyn, and now that you guys know a little bit about my title, I'd love to share with you that research today, which is on the bearing behavior of an invasive mud snail and the influence that it might have on detectability during monitoring and surveying. So as global connectivity continues to increase, we're seeing a rise in species introduced to new areas. Marine invasive species in particular tend to have traits like rapid reproduction and high dispersal abilities that allow them to establish quickly and spread across large areas. And this can have major ecological consequences. Because of this, effective monitoring is critical, not just for detecting these species early, but for understanding the impact that they might have on the ecosystems they invade. But monitoring isn't always straightforward. One of our main challenges we face is accurately estimating how many species or how many individuals are actually present, and many of these species display behaviors that might make them harder to find and count. And while we know these behaviors do exist, they're not consistently factored into survey methods. And this leaves a major gap in our ability to reliably monitor and manage these invasive species. So to explore how behavior can impact population density estimates, I focus on the marine invasive mud snail called Batellaria atramentaria. They are a cone-shaped snail, and they have the potential to reach crazy high densities within the intertidal. Originally from Asia, they were introduced to North America in the early 20th century, likely coming over with the Japanese oyster, and which were being brought over to North America to farm. The snails are basically hitchhikers on these oysters, either on the shells themselves or in the water that was used to transport them. Currently, they range from BC all the way down to California. High densities of these mud snails are a concern as they can compete for resources with native species, potentially displacing them, as seen with the native mud snail in California. Additionally, their presence may facilitate the establishment of other invasive species. In terms of habitat, these mud snails are abundant in shallow tide pools, but during the summer, they can be found across much of the intertidal, usually in muddy substrates, but also rocky and sandy areas. So earlier I mentioned how important it is to consider behavior that might make a species more cryptic when conducting density surveys in order to estimate accurately. And for example, behavioral traits like how deep an animal buries itself can directly impact detectability and potentially lead to underestimations if it's not accounted for. In the context of this mud snail, they buried themselves headfirst into the sediment using a corkscrew-like action, which is thought to prevent desiccation, but could also be a response to temperature change. What's still unclear, though, is how often they bury, how deep they bury, and how this behavior can vary across temperatures and seasons. And these gaps in our understanding matter because if snails are underground during the surveys, we may be underestimating how many of them are actually present. And understanding how these types of cryptic behaviors can impact population density estimates for invasive species is extremely important, and this is what's driving my 2 research questions. First, does burying behavior impact detectability, and does temperature change burying behavior? Let's get into the first question. So most of what we know about this species is from fieldwork that has been done in the summer. And with low tides during the day and higher temperatures, who wouldn't want to do their field work in the summer here in BC? But by only surveying in a small window of the year, we might not be getting a big picture as to what's going on with density and behavior in burying with different temperatures. So to contrast this glorious summer day, I'd like to show you a picture of my fieldwork from our February survey. And with low tides at night and lower temperatures, this might not make for the ideal study site. But my hope was to gather data that might help us understand how densities and behavior change throughout the year. Now, not all 3 of my surveys looked this grim. We had some nice weather as well. Our field site was at Rocky Point in Port Moody, which is an intertidal mudflat. And so now that you know a little bit about my main areas of focus, I'd like to tell you the methods that I used to gather my data in hopes to answer my research questions. So I used the classic transect quadrat method, and then I completed surveys once in February, March, and April. There were 3 transects laid perpendicular to shore, starting from a fixed concrete wall that served as our reference point near the high tide line. The lengths of transect varied from 39 to 99 meters, and along each transect, I placed a 25 by 25 centimeter quadrat every 3 meters alternating sides of the transect. At each quadrat, we started by counting all the snails that were on the surface of the sediment. We measured up to 10 of them and then removed them. Then we repeated this again for partially buried snails, and then we went digging for these snails in the quadrat and repeated the same measuring methods. So I also wanted to see if there was an effect of quadrat size on density estimates. So I did this by placing a 1 meter by 1 meter quadrat over the 25 by 25 centimeter quadrat and just counted all the surface snails in that quadrat. So now before I get into my results, I'd like to share with you what I thought we would see. Now I thought there would be more snails buried in cooler months, so fewer on the surface of the sediment, and as we move into warmer months, more on the surface and even more so by April. But what did we actually see from our field observations? Well, I made this plot to show snail density over the months, and on the y-axis we have snail densities, which is individual per meter squared as a function of month. So here we are seeing maybe more snails as we go through the months, but we didn't find a strong statistical evidence of this, as you can see with the large overlap of the 95% error bars. Okay. This plot is showing total snails per quadrat as a function of distance from the wall. And as we can see here in the 3 months, the distance from the wall increases, we're seeing a significant decrease in the total snails per quadrat. From 0 meters from the wall, we found more snails in April than February and more snails in March than February, but no difference between April and March. But this went away as we moved away from the wall. At the mean distance away from the wall, which was about 39 meters, there was no significant difference in total snails per quadrat across all 3 of the months. Okay, this plot is showing proportion of snails as a function of depth category. So surface was just all the snails that were on the surface, and partial was the ones partially buried, and fully buried in full. And this is of the snails that were present. So you can see there's a lot of overlap with all the error bars across the months, and the only result that is significantly different from each other is the proportion of snails on the surface in April compared to the proportion of snails that were fully buried in April. And this is showing how I wanted to see if there was a difference in quadrat size, that density of snails. And you can see that there are, well, it's hard to see here, but there actually is a larger density estimate in the small quadrats than the large quadrats per meter squared. Okay, this plot is showing total count as a function of surface count per quadrat, and the points are basically a ratio of snails over total number of snails per quadrat. And I added that black dotted line on the 1-to-1 line, which basically represents the total amount of snails per quadrat are the same as surface count, which means all the snails would be on the surface. So you can see all 3 of the months are above the 1-to-1 line, so there are snails that are buried. And then I wanted to see if these lines became closer to the 1-to-1 line, and I used the sum of surface snails and partially buried snails. And again, they're all above the 1-to-1 line, but a little closer. So back to the main research question of, does bearing behavior impact detectability? And I'm going with maybe. We'll get more into this later. Now on to the second question. To supplement my field data, I wanted to run a lab experiment to isolate the effect of temperature on bearing. And the snails and sediment were both collected from Rocky Point, the same field site, same as the field site, and brought up to SFU. So for this experiment, I had 125 snails that were all randomly placed in 5 per container. The 25 containers were randomly placed in one of the 5 different treatment temperatures. So we have 4, 8, 12, 16, and 20 degrees. And I left them there for 24 hours, checking on them periodically. And then again at the end, where I determined if they were buried or not. And if they were buried, the depth to which they buried to. And I predicted that as temperature increased, fewer snails would bury. And if they did bury, they'd bury at shallower depths. But again, what did we see? This plot showing the proportion of snails that buried as a function of treatment temperature. And there's significant overlap with the error bars again, which shows no significant difference between the proportion of snails that buried across all different treatment temperatures. This plot is showing of the snails that did bury, were their depths any different? And again, there were no significant differences between the depths they buried. So does temperature change burying behavior? My answer is also maybe. The pairwise comparison was not significant, but the initial model that compared 12 degrees to 4 degrees was significant, as there were no snails that buried in 4 degrees. And although my hypothesis was that they would bury more if they were cooled, from a metabolic standpoint, it makes sense as activity levels are lower in cooler temperatures. So let's circle back to that first question and dive into a little bit more why I said maybe. The reason why I said that bearing behavior might affect detectability is because although we might not have seen bearing as a response to temperature in the lab experiment, we still know that snails are burying because all the lines are above the 1-to-1 line. So I would say that it's still important to dig for them. And if you don't dig for them, it might impact the density estimates. So at this point, remember to bring your shovel to count these snails if you're going to. Further, although evidence was weak, there were more snails counted in April than February, meaning we are seeing fewer snails in cooler months. I also think this trend would become more apparent if more surveys were conducted. And because of this density increase, I wouldn't recommend comparing mud snail density data from winter to summer, as it might look like they're increasing rapidly, but this might just be a seasonal pattern within their density. And I hypothesized that the reason that we would get smaller estimates in cooler months would be because they are bearing, but we accounted for this in our methods. So it might not be temperature that's causing them to bury. And this might change how we think about our snail data, but it still leaves us with 2 main questions. Where are they in the winter? We checked if they were buried in the sediment, but that's causing the underestimation, but this wasn't the case. And then where are they coming from? How are we getting density estimates that are 9 times higher in April than in February? I have some ideas, but I'm not really sure. One of my ideas is that they are known to reproduce between March and July, so it could be snails coming to the surface or congregating. to spawn. Another idea is that snails are crowding up, making it seem like there are more snails in April. And overall, my field conclusion is that winter estimates are not an accurate prediction for summer estimates, or summer densities. So from this experiment, we found that it might not be temperature driving bearing behavior, which wasn't what I thought was going to be the outcome of this experiment, but an interesting result nonetheless. The lack of temperature sensitivity could be a contributing factor to why they're doing so well as an invasive species, as invasive species are known to tolerate wide temperature ranges. An alternative hypothesis to explain bearing behavior could be a response to desiccation or immersion time, or maybe even a response to predation. But we didn't see a super significant result for the incubator experiment, but we saw a hint of something. And it might be interesting to go back and look at this with higher replication. So overall, surface counts alone may result in an underestimation of mud snail density. So continue to dig for them during your surveys to improve detectability. And smaller quadrats tended to produce a higher density estimate, likely due to their clump distribution. So choose quadrat size carefully based on site characteristics and research goals. Mind the season. While bearing behavior didn't vary significantly with temperature, more snails were visible on the surface in April. And seasonal timing may still play a role in detectability and should be factored into monitoring protocols. In conclusion, accurately estimating densities of invasive species are crucial, but so is understanding behavioral patterns. For invasive species like this mud snail, monitoring efforts should integrate both biological and methodological awareness to improve early detection and long-term management. All in all, I learned many lessons from this project. It was my first project of my undergrad, and I learned a lot by trial and error. But one of the main things I learned was how important it is to think about every detail before you go into the field, and working in the intertidal, you're working against the tide. So it added another challenging layer. But thank you so much for listening to my presentation, and I'm honored to be here. And that wraps up my presentation. Thank you all for listening.
Thanks very much, Jocelyn. Any questions for Jocelyn? Sure. Do you want to start with us here, Kim?
Yeah, we have a question from Amelie. I might have missed it, but I saw 2 locations shown in your map, Vancouver and Kelowna. Could you please clarify what these locations were for? I thought they were your sampling sites, but Kelowna wouldn't be marine or estuarine.
Yeah, I was just putting perspective to where we are now and where my research site is. So my field site was only at Rocky Point down in Port Moody, not Kelowna.
Carla D'Antonio at University of California. I just wondered, when you were out there surveying, if you saw other organisms and recorded them, especially when you were burrowing and or looking in the mud, and whether or not there were any correlations between your snail abundances and the abundances of any other organisms.
Yeah, that's a great question. We definitely saw quite a few different organisms. In my project, we didn't count any of them, which is a shame, because that is a really good point. But thank you for your question.
Thanks for your talk. Stephanie Green, University of Alberta. I wondered, I might have missed this, but how did you choose the upper bound of your temperatures for the experiment?
Right. Yeah, it was deliberation with me and my lab team and Isabelle Cote. We chose those temperatures because that's the range that they would experience from like the time that I did my field work. So kind of like from January to April.
Okay. All right, let's give Jocelyn another warm round of applause. Also have some fond memories painting snails. All righty. So yes, a slight change in the agenda. We've got Dr. Vicky Maloney up next. Unfortunately, Dr. Ensing was ill and unable to join us, but anyhow, we move along. Our final presenter in the session today is Dr. Vicky Maloney from Enview Canada, where she's the Solutions Development and Tech Services Manager. Since joining Enview in 2022, she's dedicated herself to addressing the challenges of invasive species management and the responsible use of chemistries. In her current role, she
In her current role, she collaborates closely with customers, landowners, fellow researchers to strive to develop innovative solutions for effective land management and invasive species control. All right.
Thank you.
Take it away, Peter. Sorry.
Okay. Okay, so this is a little bit of a departure from snails, but we're going to switch over to invasive annual grasses. And so this, so yeah, like you said, my name is Vicki Maloney. I'm a research scientist for Enview Canada. So what I'm going to talk about today is a chemical called indaziflam, and it's used for invasive annual grass control in both the US and Canada. And then I'll talk as well about some of our results on the restoration of biodiversity in these rangelands that have been treated operationally with this chemical in the Western US. This work was conducted in collaboration with multiple cooperators across Canada and the US, as well as my colleagues in the US, Dr. Shannon Clark and Dr. Harry Quick. Okay, so before I dive into kind of the projects, I just wanted to set kind of the stage of what we mean by invasive annual grasses, as some people might not be aware of what they are. I mean, as the name suggests, of course, they are invasive non-native species. They complete their life cycle in a single year. But they also have the ability, like most invasive species, to spread really, really aggressively and quickly beyond their native range, often outcompeting native vegetation. These grasses establish quickly, produce a lot of seed, and typically germinate in the fall. And I say typically because we've noticed within, especially in the more northern ranges, these can be kind of adapt to the different ecological conditions. So typically they would germinate in the fall over winter as a seedling, and then grow the following year. But we've seen variations in that habitat or that growth pattern, depending on the weather and the climate in those regions. In the US, the worst invaders are downy brome or cheatgrass or Bromus tectorum and Medusa head right here, kind of got this unique pattern, and then ventonata. In Canada, by far the worst invader is downy brome. It's not a new species to Canada, but it's spreading quite rapidly. We do not currently have any reports of Medusa head within Canada, but we do have reports of ventonata in Canada. So these are something we really want to keep track of moving forward. We also have Japanese brome in Canada. So looking at the spread of these grasses across the Great Basin of the US, we can see that there has been over 10-fold increase since the early 1990s. So this was a 1990, and this is 2018 is the latest data we have from this. And you can only imagine how much worse it's gotten since then. And you can see this is the spread of the invasive annual grasses and the 10-fold increase in those invasive annual grasses. And this is particularly Bromus tectorum, and we talk about an undesirable stable state, and that's kind of what you mean by it's greater than 50% Bromus tectorum is considered an undesirable stable state. And it's really correlated when we look at forage performance across the US rangelands. So with this introduction of this undesirable forage taking over almost 230,000 hectares per year, we're getting, of course, a lot of reduction in the performance of forage across these US rangelands. If we look more closely, specifically at Canada, we see that the major infestations are coming from the south. They're coming from across the border, but they are spreading northward. And with climate change, the leading edge has moved upslope from about 138 meters to 459. Of course, this varies by ecoregion and aspect, but we are seeing that trend in the increase in the spread of these invasive annual grasses into Canada. And I want to make kind of a caveat here with this map is that we have really, really poor mapping and reporting of these invasive annual grasses in Canada. And grasses are typically very, very hard to identify, as you probably most know. And so we're not getting just the layperson reporting that they see these grasses. They just look like typical grasses, and it's not as obvious. So again, we really need to be able to increase this reporting in Canada. Okay, so when it comes to controlling these invasive annual grasses, the main chemical that is currently used in the US is indaziflam, and it's marketed in the US as Rejuva. In Canada, we have it as Esplanade for use in industrial vegetation management, but Rejuva in the US is used in range and pasture and grassland settings. And it is a group 29 herbicide. It's categorized as a cellulose biosynthesis inhibitor. And it works in the top layer of the soil and inhibits weed seed germination in that top layer of soil. So these desirable perennial species that are deeper rooted, can avoid the herbicide uptake and grow through that herbicide layer, where these annual grasses, which of course are very shallow rooted and have a large seed bank right in that upper top of the soil, are the ones that are majorly affected. And here we can just see with increasing rates of indaziflam, we are seeing this reduction in seed germination as well as root growth. And you can see here how the actual cell wall is being affected by the indaziflam. We have conducted many small plot trials with several cooperative in the Western US, and we've consistently seen that indaziflam does indeed provide long-term invasive annual grass control. The particularly results are in combination of, these particular results are from 10 different studies that all show that 4 years after treatment, we're still seeing over 90% control of annual grasses. And this is just in comparison to imazapic that they also use in the US sometimes for invasive annual grass control, not a chemical that we will currently use in Canada. But you can see that this is 4 years after treatment, we're still getting almost close to 90% reduction in the invasive annual grasses in these ecosystems. And equally important, of course, is that we see the number of native plant species, including the annual forbs and other rare species, that are increasing in these treatment plots. So you can see in the indaziflam treatment plots, we're seeing increase in shrubs, warm season grasses, cool season grasses, annual forbs, as well as rare species in these treated plots. So that was just to summarize a lot of data in just a couple of slides, because that's not really the main point of this presentation. The main point is to talk about what's going on in Canada and what's happening with the biodiversity. So in Canada, we have seen where it's not this spread that it is to the extent that it is in the US, but where we've seen fires go through, and this is an example from outside Waterton Parkfront, where in 2017, a huge fire burned about, it was about 16,000 or 19,000 hectares within the park and about 16,000 hectares of the surrounding rangeland. And what came back was almost near monocultures of cheatgrass downy brome. That's what this is here. And so where it might not seem like it's present, given the right conditions, it can quickly take over an ecosystem. And so we conducted some Canadian-specific trials in this area in order to look at how indaziflam can perform in these Western ecosystems. And we were able to see that indaziflam and its tank mixes, anything that contained indaziflam, significantly reduced the Bromus tectorum cover. And in this case, we applied in August and October, and we can see again the difference in that germination timing. So if it's pre-germination, which we tend to see in August, you're going to see that. So again, in the 1 year after application, in the August timing, we're seeing a reduction. Whereas you're not going to see that reduction if it was applied in October, because you've missed that pre-germination window. But 2 years after application, you will see that substantial reduction in cover of the invasive annual grasses. And then this was just to compare tank mix partners with commonly used herbicides. This one is True Range. It's for a broadleaf herbicide. And then Plateau is a herbicide used in the US again. But again, we're seeing a substantial reduction in that herbicide, in the annual grass cover in these areas, just to substantiate that we do see the same results in Canada as we do in the US. Again, we're excited to see that with all of the treatments that included indaziflam, there was a significant increase in the native grass cover, and that continued into the second year after treatment. We also saw that the True Range tank mix partner, that one that's a broadleaf herbicide, was really increasing, even seeing further increase in that native grass more than with just indaziflam alone. So this is the indaziflam alone. We see an increase, but again, with that tank mix partner, we're seeing even further increase in the native grass cover in those areas. We also supported a master's student at the University of Alberta, Annabelle Dumbro, and she recently published this paper showing, again, similar results, that indaziflam does indeed provide long-term reduction of annual brome grass in Canada's mixed grass prairies. And so if you're interested, you could look up that paper. It was published in Rangeland Ecology and Management last year. So this gave us a really good predictive that we are seeing an increase in biodiversity in these treated sites, but we wanted to kind of look at a bigger scale and see what really is happening in larger commercially operationally applied treatments of indaziflam. So there was multiple different sites across the US that we looked at, one in Nebraska, one in Oregon, and one in Wyoming, and all different kind of ecosystems, different invasion, where they were in the invasion pattern. But I'm going to talk specifically just about the Wyoming one because it's more similar to what we're currently seeing in Canada when it comes to the spread. So this is what the sites looked like. It was this is an example of a paired non-treated site that was treated with indaziflam in August of 2022. And this was treated. This is the non-treated site. And then this is 2 years after application. And this was applied specifically just with indaziflam, not with that tank mix partner plateau that they tend to use in the US. So it's a little bit more applicable to what we will be seeing in Canada applications. The data was collected using the Guide to Rangeland Monitoring and Assessment. We collected a bunch of different data. I'm just going to specifically, for the sake of time, just talk about dry weight rank. So this is data pre-treatment. We can see that this site was not completely degraded. It still had substantial cover of native grasses, but there was also this Japanese brome and the ventonata, which are those invasive annual grasses. And sorry, the arrows are a little bit messed up here, but anything that has an orange arrow means that it was increased in dry weight rank in the non-treated versus the treated, and vice versa for the blue arrows. So fast forward to a year after treatment, we're seeing that the annual grasses, so that being the ventonata and the Japanese brome, have these orange arrows. That means they're significantly increased in the untreated versus the treated. And we are seeing, first of all, an increase in plant species and biodiversity in those treated plots that we're not seeing in the untreated, but also a large increase in native species like the wheatgrasses and the big sagebrush in those treated sites. And then one more year, this is 2024. And while we continue to see, shifts in that plant community, which you would expect from any sort of natural environment, we're still not seeing any of those invasive annual grasses, in this case, the ventonata and the Japanese brome, in those treated sites. And we are seeing even a further increase of those grasses in the untreated sites. And this, grass are wonderful and they show data, but I think that it can be visualized even better if we look at it from like a larger scale. This is non-treated versus treated 3 years after treatment in Scottsbluff, Nebraska. Again, large, you can see the biodiversity that's returning after treatment. This is in Kurt Gowdy State Park in Cheyenne, Wyoming, Cherokee State Park Wildlife Area. So you really can see that there's a large increase in biodiversity after the treatment of these sites that have a near monoculture of invasive annual grasses. So what are the lessons learned? Indaziflam does indeed have the same efficacy of control of invasive annual grasses in Canadian mixed grass prairies as it does in the Western US. The biodiversity monitoring in these operationally treated lands showed that every region had lower invasive annual grass frequency in indaziflam treated plots. So if we look specifically at the percentage of invasive annual grasses, they made up about between 17.5 and 37.3% of species composition in the non-treated sites, whereas in the treated sites, it was almost non-existent, sometimes being up to about 3.7%. And then if the dry weight rank increased in all of the native grasses increased in all of those treated sites. And then the last thing I really want to say is that the current state of annual grass infestation in Canada is really not well understood. It's a new problem. People aren't necessarily talking about it to the extent that they maybe should be. So it needs to be better understood and recognized. So with that, I will take any questions.
Hi, I'm Em Lim from Simon Fraser University. Very interesting talk. Something you mentioned at the beginning stood out to me. You said that grass is obviously very hard to identify. Your average layperson isn't really reporting them. I was curious, actually just looked it up real quickly, but there's quite a few sightings of them on the platform iNaturalist.
Yeah.
And I was wondering whether like citizen science or like community science platforms such as iNaturalist are being used at all in terms of invasive tracking. I know BC has like plant specific portals that you can go into and say that you've seen something, but I'm wondering if like these more general ones are getting used at all.
I think they are. The data that I showed was from EdMaps. So similar kind of thing. It's not necessarily, scientists that are identifying it and doing that. There are new ways of detecting these annual grasses that are developing through detection from satellite and that sort of thing. And I think that's really going to advance it because again, like iNaturalist, there's apps that are good, like, that you can identify grasses using an app, but those are decent. But people who are just walking down a path, they see like a hawkweed or like something like that. It's obvious to them, this doesn't belong here. It's not the case with the plants. They really need to be looking for them to know that they're invasive. So if you know what to look for, you can find it. But if you don't know what to look for.
Yeah. Do you think like like hundreds or like almost like over like 1,000 observations and are people that just see a grass and post it and then an expert on iNat would then identify it?
Yeah.
Or like why do you think they're just posting random grass photos and then someone else is coming through?
For EdMaps?
No, for like iNaturalist, like programs for like random people.
People are actually experts are going in and identifying.
Yeah, I think there's a couple of folks. There's like,
Okay.
I don't know if you know Barbara Wilson. She's like Sedge Queen on iNat, but she's like one of like the grass experts, I guess. But she's like quite active on there. And
well, that would be great if people can do that more. Yeah, we need that. Yeah. Oh, right.
Hi, this is Rebecca Nelson from Utah State. Great talk. Thank you. I'm really curious for the series of plots you were monitoring throughout Western North America, if the increase in the native forbs from the annual grass removal, if you noticed any downstream effects on pollinators or other insects. Thank you.
Yeah, so we have done research on pollinator visits, and we did see increases in pollinator visits and specifically with the native forbs returning. Yeah, so we have data on that. We also have data on a mule deer and elk habitat and improvement of mule deer and elk habitat, actually looking at visitations in sites, non-treated and treated sites, and seeing an increase in those, larger herbivores as well in these sites. So yeah, both. Yeah.
Carla D'Antonio from University of California. Thank you for your talk. It was encouraging in a lot of ways. We certainly have a ton of annual grass problems where I live. I'm curious about whether your increase in perennial grasses that you reported in your last series of slides was due to growth of existing ones or recruitment of new individuals, since the herbicide is a pre-emergent, like you would think it would have a negative effect on all seeds and
it does.
Okay.
It does, yeah. So that recruitment would be from rootstock for the most part, yeah, for sure. Except for like when we're looking at 4 years after, then you can see you can see some sort of recolonization of seed as long. But if you've removed that seed bank from the cheatgrass, then you're not getting the cheatgrass coming back necessarily if you just get recolonization of native seed. But originally, when you see the return of the perennial grasses, it is from rootstock. And that's why we really don't want to get to the degraded state that we have seen in some parts of the Great Basin, because then when you're killing the cheatgrass, you're getting bare ground because you're not getting anything to come back. Fortunately, in Canada, we're not at that state. We're more in a, we have to act now, but it's not necessarily complete monocultures of cheatgrass yet. We'll still get recruitment from the native species. Oh, sorry.
Chandra Moffat, Agriculture Canada. Thanks so much for your talk. Invasive grasses are something that we worry about a lot. And from a biocontrol perspective, there's few herbivores, more potential for pathogens. But I just wanted to follow up on Lim's comment about data sources. So something that we do a lot in my group is look at, different data sources to do our mapping. And one thing that I've definitely noticed is that EdMaps is really not used almost at all in Canada. So there's a real lack of records in EdMaps for a lot of invasives, but a couple resources like in BC in particular, there's really strong efforts to put data into iNaturalist. There's universities and BC parks that have large scale projects, and those are research grade observations, and you can filter by research grade in iNaturalist. So there's a wealth of verified data there. But another more local resource would be Invasive BC, which is run by BC Ministry of Forests and the Interministry Invasive Species Working Group. And there's more and more invasive grass records in there as well.
I know BC has been, I don't know if they're better at reporting, but ventonata is definitely more prevalent in BC than it is in Alberta. And it doesn't necessarily make sense that it would be. And I just think maybe BC is better at reporting it.
There's definitely a large scale effort to document the distribution, and especially for something like ventonata, like early detection, rapid response. So though, if you're interested, like those data sources might, give you a better picture of the known distributions, at least in BC. And it's a problem across Canada that we don't have something like EdMaps that's like one, harmonized resource for records for invasives. And in BC, we do a great job, but then it's hard at the national level. And that's something we run into a lot, is using like GBIF and EdMaps and INAT and Invasive BC and like as many different databases as we can. But there's a lot of gaps. So appreciate that too.
Hi, Nicholas Yvon from UBCO in Agriculture Canada. I think you briefly touched on this answer, but I'll ask it more directly. You showed 2-year and 4-year decrease in Bromus tectorum, and I was wondering if that's a persistence in the herbicide or if that's an increase in community competition that's outcompeting potential germination later on, like especially in year 4. Is there herbicide left or?
So I would say it's a mix. It's a mix of both. It is a residual herbicide. It does have a half, we say 8 months, but we've seen longer depending on environmental conditions and, the microbial community. It's mainly broken down by microbes, not phytolysis or those sort of things. So, it's a mix of both. When I show 4 years, I doubt there's any chemical left in those 4 years. That's more because the seed bank has been eliminated, because it will just keep on trying to regerminate and then it won't be able to, and then it will try again and it can't, and then eventually there's no seed left. So, yeah.
All right. Hey, thank you so much, Vicky. Let's put our hands together again for everyone. Nicole, Jocelyn, and Vicky. Thanks so much. Okay, well, it's been a great first day. It's not quite over yet, but there's a little more fun to come. I'm really encouraged that we've heard a lot of diverse presentations today from different taxa, different landscapes. We heard that collaboration is pretty key across a lot of our work, and especially hearing a lot about a lot about new tools, innovation, and technology. Indoor ecology as well, which is a new term. And it's great to see such a age range, not age range necessarily, but experience range from folks with 40 years down to Jocelyn, who's just starting her research journey. So that's really awesome that we're seeing, especially some of those early career researchers. So yes, it's great to learn, but it's also great to network. So we've got two fantastic opportunities to get to know each other a little more. So if you're joining us on the field trip, the bus is leaving from lot 4. So you're just going to go out past that, the admin building. You'll pass 2 more, and then lot F will just be over the pedestrian walkway. So there should be an obvious big bus sitting there. Let's see. We also need to thank all of our sponsors today. So yeah, none of this stuff could be happening without their support. And we'll get a chance to meet and mingle with all the sponsors at 5:30 in the poster session and the reception. That again will be over towards that parking lot. It'll be in the University Center ballroom. So that'll just be perpendicular to that lot F. Let's see what else. There is a quick survey. If you're sticking around, don't worry about capturing this now. And for those online, you can follow the link at any time, but always keen on hearing what you think and how to improve our conference 3 years from now. Yeah, tomorrow we will, for those of you who are not joining us on any of those events, 8:30 start tomorrow. Our keynote speaker will be Dr. Green from University of Alberta. And yeah, very excited to hit day 2. Thanks so much. Pardon me? The bus is probably there right now. Yeah, the sooner we can get on it, the better, and get to that winery and do some sampling and learning, obviously. Alrighty. Well, thanks so much. And yeah, thanks to everyone online. Hope it all came through well today, and we'll catch you all tomorrow.